become the virtual format that we've become used to over the last year and a half has allowed for this kind of workshop to take place, where we can have a focused topic and allow more participation from people across Canada, including students who may not be able to travel to in-person meetings. So look for more of these workshops throughout the year. So in particular, I helped organize this session with Amy Fungi of the Ontario Ginseng Growers Association and Andrea Bottero of the University of Calgary. And we deal with some of the more difficult to manage pathogens. So that's why we organized this workshop today, to kind of improve our understanding, monitoring and management of soil-borne pathogens on a diversity of crops and cropping systems across Canada. So I work closely with ginseng growers, so I know the challenges of managing these pests from the grower perspective, as well as conducted a lot of research with Amy on ginseng diseases. We deal with a lot of the typical genera of pathogens, including Phytophthora, Pythium, Fusarium, Razoctonia, Verticillium, both root knot and root lesion nematodes. And we also deal with a very unique challenge to ginseng, which is ginseng replant disease caused by another soil-borne pathogen, Eleonectria morse panacis. From the grower perspective, we've done a lot of research on fumigation, alternatives to fumigation, amendments, fungicides, and we know the practicalities of implementing these techniques and also monitoring for these pathogens. Particularly, our growers fumigate, and fumigation, although it should work in theory, there's practicalities around getting the entire soil fumigated without leaving a refuge of pathogens to reinfect the soil, and the issues around obviously controlling everything in the soil and including the beneficials. With fungicides, there's issues getting it down to where it needs to go. Our growers apply fungicides regularly, but we've learned that if you do not apply them, a lot of the fungicides, if you don't apply them during a rain or with rain or irrigation right afterwards, they stick like glue to the straw and will not go any further down to the root zone. So it is a challenge for growers to be spraying 30, 40, 50 acres of ginseng and getting irrigation on before it dries onto the soil or onto the straw. And at the same time, we know there's ginseng's well, normally a high value crop, not so much lately, but our growers can afford a lot of these techniques, like $2,500 an acre for fumigation is well worth it for them. Where we know across Canada, there's other crops with much lower margins where you can't be affording fumigation and additional regular fungicide applications. And there's also practicalities around growing season for, especially on the prairies, where fumigation and some alternative techniques would cost basically mean a year without production. So we tried to organize this conference to represent a diversity of crops across Canada, from very high value crops like cannabis to much lower value crops with lower margins. And we hope today's workshop will be very interactive. So feel free to chat back and forth with fellow participants and with the speakers in the chat. We ask that please keep your cameras off and your microphones off unless you are speaking, with the exception of the question and answer period that's at the end of the workshop, where we hope people can turn their cameras on and we can have a thorough discussion on the topic. If you have a question for a particular speaker, either enter it into the chat or raise your hand, and we'll try to do those in succession. If we don't get to them in that time slot, we can reapproach them at the end. Also, we have a lot of experts on the workshop today who may not be speakers, and feel free, if you have something to contribute to the discussion in the chat, to enter that. and also during the question period. So with that, I'm going to pass it on to our morning session chair, Amy Fung-Shi. Amy is a research associate with the Ontario Ginseng Growers Association and also a PhD candidate in the School of Environmental Sciences at the University of Guelph. And I'll pass it on to Amy to introduce our first speaker.
Thanks, Sean. So I'm Amy Fung-Shi. So I'll be chairing the first session, which we have 5 speakers. So the full agenda and the full bio of all the speakers, you can find them on the CPS website, which is providing the link that you receive along with your Zoom link. I will remind all the speakers when there are 5 minutes left for your presentation. And after the presentation, there are 5 minutes for questions. If you don't get time to address all the questions, we can wait until the final general discussion period at 3:25 Eastern Time. So I'll stop sharing my screen. And then the first speaker is Dr. Steve Struckoff from University of Alberta. He has been working on the clubroot for a very long time, and today he'll talk to us about clubroot, a soil-borne challenge to canola production.
Thank you. See if I can share my screen here. Okay, share. Let me see. Can you guys see my screen? Hopefully you can.
Yes, we can.
Okay, great. Okay, and there's my little, whatever it's called, that little dot. Anyhow, pointer, sorry. Okay, thank you very much, Amy, for that introduction. And thank you for the organizers to inviting me to speak a little bit about clubroot. So before I begin, I'd just like to briefly acknowledge the people who have contributed some of the information I'm presenting today. So my name is indicated is Steven Struckoff. I'd like to acknowledge Victor Manoli, who's a technician here at the University of Alberta in our group, Keisha Holman, who's a PhD student working with myself and Dr. Xiaofeng Huang, Michael Harding, who's a plant pathologist with Alberta Agriculture and Forestry, and Dr. Xiaofeng Huang, who's a professor of applied plant pathology in our department here at the U of A. So this is an outline of my presentation. Just start off with a brief introduction to clubroot. I suspect many people in the audience are quite familiar with the disease. Then I want to move to talk about clubroot management and genetic resistance. So really, I'll probably focus more on genetic resistance. Then talk a little bit about the emergence of new pathotypes of the clubroot pathogen and the release of cultivars that carry so-called second-generation resistance, and a little bit of what we're finding there, and finish off with some conclusions. So clubroot is a soil-borne disease of crucifers caused by the obligate parasite Plasmodiophora brassicae. It attacks the roots of susceptible hosts, causing the formation of these large galls or clubs on the root system. These galls interfere with water and nutrient uptake, resulting in severe yield and quality losses, as illustrated in this image. The pathogen also exhibits what we call physiologic specialization, meaning there are different pathotypes or races that differ in their virulence or ability to cause disease on specific host genotypes. On canola in Western Canada, clubroot was first identified in 2003 in 12 fields in central Alberta. They were found, this is Edmonton right here where I have my pointer. So they were found just around the Edmonton area. But surveys over the last 17, 18 years have shown, as illustrated by this map, that the disease has been spreading across Alberta, and it's now found in 44 or 66 counties or municipal districts where canola is grown. It's also increasing in Saskatchewan and Manitoba, and as well as in North Dakota. So it's become really a prairie-wide problem. The management of clubroot relies on, on several strategies. The initial strategy that was recommended to manage the disease was to sanitize field equipment. So washing off machinery to remove infested soil from the equipment so we wouldn't move it from field to field. Another strategy that was initially recommended was to use longer rotations out of canola to prevent or slow down inoculum buildup. Unfortunately, while these strategies are valuable, there's been limited adoption by a variety due to a variety of reasons. So they haven't been as widely used as maybe we would have liked, and this is kind of reflecting the fact that the cases of clubroot have continued to increase since its initial identification. Chemical control and soil amendments have also been evaluated as strategies to manage clubroot. And many of these are derived from work with cruciferous vegetables, and there's been efforts to try to apply them to canola cropping systems. So things like fumigants to eradicate localized infestations, the application of hydrated lime or limestone to increase the soil pH, which the clubroot pathogen doesn't like, and thereby try to reduce disease levels. Unfortunately, none of these methods have really been widely adopted, again, for a variety of reasons. The cost, in many cases, it can be prohibitively expensive for canola cropping systems. In some cases, it's not really practical. In the case of the fumigants, there may be undesirable environmental effects. And in some instances, we don't get the greatest of results. They're kind of mixed results or variable results in terms of getting sufficient control of the disease. So given these limitations with sanitation, cultural strategies, and chemical control and soil amendments, there have been significant efforts to develop canola varieties that carry genetic resistance. So clubroot-resistant canola was first introduced onto the Canadian market in 2009 and 2010. So the first variety came out in 2009, and followed by several other products in 2010. And there's been new varieties introduced nearly every year. And these varieties have had excellent resistance to all the pathotypes of the pathogen that were known at that time. This, combined with the fact that they were found in that they came in an overall very good package, very good yielding, kind of elite lines, they soon became the most important clubroot management tool. And probably for some farmers, it was probably the only clubroot management tool that they used. Unfortunately, in 2013, so about 40 years after the very first clubroot-resistant variety was introduced, we found the first cases of resistance-breaking pathotypes of Plasmodium brassicae. So these were pathotypes that could overcome the resistance in these hosts. Continued monitoring since then has led to the identification of more fields where resistance has been overcome. By 2020, we had about 353 fields where we knew that there were resistance. We had confirmed that there were resistance-breaking strains of the pathogen present. And this represents, for Alberta at least, about 10% of the fields that we know have clubroot. We have probably over 3,500 fields that we know have clubroot in Alberta, and about at least 350 of them have resistance issues. One issue that we had with these resistance-breaking pathotypes is that initially we couldn't distinguish them from the quote-unquote old pathotypes or original pathotype based on their virulence on the host differential sets that were available at the time, and particularly the Williams differential set that we were using quite extensively, as well as the differentials of Sommet et al., which was originally developed in France. So this led to an effort to develop the so-called Canadian clubroot differential set, or CCD set, which consists of 13 differential hosts, including some of the old differentials, for instance, those of Williams, plus some genotypes of Brassica napias that are of particular interest to Canadian production. So on the CCD set, pathotypes are given a nomenclature that consists of a number indicating the Williams classification. So, pathotype 2, 3, 4, 5, and so on, followed by a letter denoting the virulence on the CCD set. So, for instance, we could have Williams pathotype 3, but we can detect multiple variants of Williams pathotype 3 on the CCD set, and we would label those 3A, 3B, 3C, and so on. And using these differentials, and through continued surveillance over the last 6 or 7 years, we've been able to identify 43 pathotypes of Plasmodium brassicae in Canada, and this is up to the end of 2020. Of these 43, 25 overcome the resistance in at least some clubber-resistant canola cultivars, and 18 do not overcome resistance. Now, although I'm saying there's 43 pathotypes, I should say some of these are very rare. For instance, the ones shown in red have only been identified kind of in the laboratory from single spore isolations made from field isolates of the pathogen. And others, maybe we found only 1 or 2 fields. So typically there's been a few pathotypes that rise to predominance or are most common. So this is an illustration from a figure from Keisha Holman's master's thesis and her paper that was published this year, but was using data collected in 2017 to 2018. So basically this let's just look at the top one. This is a pie chart showing the pathotypes found in Canada over those 2 years, sorry, in Western Canada on the prairies through testing. So the ones that have a star beside them are pathotypes that can overcome or break this genetic resistance. So as you can see, pathotype 3A was predominant, representing about half of all pathotypes identified. 3D, another resistance breaking pathotype, has been usually the second most common. 3H is one of the original pathotypes. It's very virulent on canola that doesn't carry genetic resistance, but usually can be controlled by resistant cultivars. And then there were a slew of other pathotypes that were found in small numbers. So most of the testing was done with samples from Alberta. So you can see this. So this large graph really reflects what's going on in Alberta. But there was also limited testing in Manitoba and Saskatchewan, where you can see in Manitoba in particular, there was a lot of pathotype diversity, and in Saskatchewan, it was mainly the old pathotype 3H. So this is interesting and suggests that 3A and 3D and 3H are the predominant pathotypes. However, what we've been finding is that there has been some shifts in the last few years in this prevalence. And I wanted to highlight, we haven't published this yet, but we've started to see a decline in the relative prevalence of pathotype 3A. So what was used to be the most common resistance breaking pathotype. It made up probably about 63% of all field isolates we tested in between 2014 and 2016. But by 2020, it was making up 12 to 13% of the total. Pathotype 3D, another resistance breaking pathotype, has been more or less stable at around, I don't know, 25, 30%, as has 3H, the old pathotype 3H that doesn't overcome resistance. But what we've really been finding is there's been a proliferation and increase in so-called other pathotypes. And these are various pathotypes. I wouldn't say there's one that's particularly prevalent. And unlike 3A, that even though there's only now 12 or 13%, we're still finding it fairly widespread. Some of these other pathotypes tend to be more localized to specific counties and regions. And many of these, but not all, are resistance breaking. So this just shows just one example of what some of these emerging new pathotypes look like. I wanted to highlight one that we found called that we call 8E. So back in 2014 to 2016, 3A was by far the most dominant, making up 63% of the samples. And 8E was a rare pathotype. Maybe we found in a few fields, it was about 3%. Since then, it's been increasing. And by 2020, 8E made up about 13% of what we were finding, and 3A made up about 12%. And the only thing is that these 3A was still fairly widely distributed, while 8E, we found it more localized to the Edmonton region, to south of Edmonton. And it's difficult to say necessarily. We're trying to understand what's driving some of these changes, but when we look at kind of the reactions, they're similar pathotypes overall, except that 8E, for example, doesn't cause much disease on one of the differentials, Brassica oleracea, called Jersey Queen, which is one of the original Williams pathotypes. And it also, it can attack, first generation resistant cultivars, but doesn't attack Mendel, which is a winter oilseed rape from Europe, and is believed to be one of the sources of resistance for some of the Canadian cultivars. And there's other examples of other pathotype that seem to be increasing. Some is clear what, maybe a little bit more why, others not so much. For instance, we've been finding over the last 2 or 3 years an increase in the old pathotype 8N, which as far as we know, doesn't break resistance. There's various pathotype 5 variants that are increasing, and also there's one called 9E that's a resistance breaking that's also maybe now 6 or 7% of the total. So we're seeing these shifts in a more, I guess you could say, more fragmented pathotype situation than before. Now, and like I said, many of these pathotypes do break or overcome resistance. So the resistance in most of the club-resistant canola varieties available even today, but certainly before there were any issues with resistance, appears to be derived from this European winter Brassica napus or rapeseed cultivar Mendel. Now, retroactively, we often refer to this as first generation resistance because it was a resistance that was first introduced. However, since the emergence of new pathotypes, there's been really a concerted effort to develop canola with so-called second generation resistance. And this is kind of a generic catch-all term to refer to varieties that carry different types of resistance. So it's potentially an important new tool for club root control, but the source or sources of this resistance is not in the public domain. It's likely different from first generation resistance and could include things like different resistance genes or potentially stacked resistance genes. While second generation resistance is an important tool, it's not a magic bullet. And certainly depending on the exact source or sources of this resistance and the cultivation practices, it may also be vulnerable to pathotype shifts, just as first generation resistance was. So what we've been doing over the last couple of years is collecting field isolates from fields or crops planted to second generation club resistant canola crops and collecting symptomatic plants that are developing club root in these fields and bring them back to our lab for testing under controlled conditions against a suite of cultivars that carry second generation resistance, and also for pathotyping on the CCD set. Right now, we have about 30 such isolates collected in our collection. We have results, or at least preliminary results, from 23 of these. So I just wanted to highlight a few examples of some of the, 23 isolates, and they're not all behaving the same, but just to show you what some of these things are looking like. So a couple of examples. So this is, for example, testing we did with a field isolate called BL319. This was recovered from a second generation club-resistant canola crop in Alberta in 2019. And when we pathotyped this, it came out as the resistance breaking pathotype 3A that could overcome first generation resistance. So here this graph shows how some of these different host genotypes react to it. So this is on the y-axis, we have the index of disease, 0 meaning no disease, 100 meaning completely diseased. ECDO2 is a Brassica rapa that seems to be, it's included as a check, seems to be resistant to everything we've ever tested with it from Canada. Then we tested a first generation club resistant check, which would expect to be susceptible to pathotype 3A. And then we tested 8 different canola cultivars carrying so-called second generation resistance. And then we repeated the experiment twice. And as what you can see is that the vast majority of the second generation club resistant cultivars were susceptible, either moderately or very highly susceptible to this field isolate from this field. There was one cultivar that seemed to be holding up well against it. We also compared the reaction of these same lines or same cultivars to a pathotype 3A control. This was also a field isolate, but collected in 2014 and never exposed to second generation resistance. Unfortunately, in this case, everything was again still most of them, not all of them, but most were highly susceptible to 3A, except again, this particular cultivar, and one of them produced a bit of less disease. So definitely, an isolate like this has the potential to cause significant issues with second generation cultivars. As a way of another example, I just wanted to show you some results from a collection of 5 different field isolates Again, all collected from Alberta in 2019, and all coming from crops from fields that had been planted to second generation clubroot-resistant canola crops. So what I'm showing here is the same information, the index of disease and response to these isolates, but we're just taking the average reaction to all these 5 isolates. But these isolates are interesting because they were on the Canadian clubroot differential set, they're classified as pathotype 3H, so as the old pathotype 3H that doesn't overcome resistance. And what we find is that most of these second generation resistant cultivars are providing good resistance against 3H, but 2 of them showed quite a bit of disease to 3H, but mind you, this came from these came from second generation clubroot resistant fields. So perhaps something had changed in that 3H. However, when we also tested these second generation resistant cultivars against a pathotype 3H control, again from 2014, that had never seen second generation resistance. And unfortunately, we did find 2 cultivars that had second generation resistance that were actually appeared to be very susceptible to the old pathotype 3H. And I hope that, when the people that as breeders and as scientists, we don't become so fixated on second generation resistance and 3A and the resistance breaking pathotypes that we forget about the old pathotypes, because they're still there. 3H is still quite common, so it still has a lot of potential to cause harm. So that was just a little bit, you're just trying to highlight a few results. So just in terms of a summary, I guess you could say so far, based on the testing we've done with 23 isolates, most second generation clubroot resistant cultivars develop moderate to high disease severities in response to at least some of the field isolates we tested. In most cases, I would say in response to most of the field isolates we tested. And 2 of these cultivars appear to be susceptible to the old and previously dominant pathotype 3H, as seen in these images. On the flip side, good news, 1 cultivar was resistant to 22 of the 23 field isolates tested. So I'm virtually out of time, so I'd just like to end off with some conclusions. Clubroot has emerged as a major disease of canola in Canada, and genetic resistance represents the most important and widely used clubroot management tool. But the proliferation of resistance breaking pathotypes is a cause for concern and indicates that the canola crop is still at risk. And really, we have to think about proper resistance stewardship to ensure the continued efficacy of resistance in the long term. And with that, I'd like to end and thank our funders, ARDAR, Alberta Canola, Sask Canola, and Western Grains Research Foundation, who all contributed to the data I presented today, as well as Alberta Agriculture and Forestry for in-kind support, and our research team and collaborators. And thank you.
Thank you, Dr. Struckoff. Is there any questions for
There are
him?
There are a few questions in the chat. First one, do you think that releasing of the resistance sources for first and second generation resistance cultivars could be useful to ensure more durable cultivars, since it could allow us to implement gene rotation for the selection of cultivars while still cropping resistant cultivars?
I think, yeah, that's a very good question. I think it has a lot of potential. The only issue, at least right now, is that we don't know, because of proprietary concerns, we don't necessarily know what's in some of these cultivars in terms of conferring resistance. So it makes it hard to have a knowledge-based knowledge to say, okay, this carries this resistance gene, this carries this one, so maybe we can rotate them. Right now, it's a little bit of guesswork, but I think there is potential there.
Okay, next question. It looks like that selection of cultivars for each field relies on the pathotyping of populations. However, how feasible is it to conduct pathotyping for farmers?
Okay, yeah, that's a good question. I would say in most cases the growers make selections based on general information, because we only test a small, even if we test 130 pathotypes in a year, or 130 samples, we still wouldn't get information for most of the field. So that information could be useful, but there's no commercially available pathotyping yet. I didn't talk about this, but we have other projects, for instance, based on other molecular technologies to do pathotyping which could potentially eventually be, maybe offered by commercial diagnostics labs and so on. But right now, most of the pathotyping is done kind of the old classical way, and that's not feasible on a prairie-wide, scale.
Okay. Next question, what are the average yield losses for growers for every 10% disease level?
Okay, oh, that's an interesting question. We have a paper by a student, Andrea Botero, who's nearly, I've been accepted with minor revisions, so we're just doing a few of the minor revisions, and she's developed kind of a yield loss model. I could say, so under field conditions, yeah, if I know this is being recorded, so I don't want to misquote it and have this saved for posterity, but it varies, but I think it was about, for every 10% decline in yield, probably about, for every 10% increase in the index of disease severity, and maybe Andrea's listening, she can correct me if I'm wrong. I think under field conditions, on average, was maybe 2.5% yield loss, something like that, under field.
Something about 2.5% for each 1% increase in the disease index.
Yeah, thank you, Andrea. Yeah, I thought about mentioning a little bit of that, but just I didn't have enough time today, so I just tried to focus it.
Okay, distribution of pathotypes in Western provinces is related to selection pressure imposed by, distribution of pathotypes in Western provinces is related to selection pressure imposed by host plant genetic background.
Yeah, I think definitely we can't say it for, but I think definitely the what we're cropping is it's exerting selection pressure on the pathogens. That's why I think we've seen these kind of shifts, and we know also from experiments that we've done under and others have done under greenhouse and lab conditions, as well as experience from other regions, for instance, vegetables in the US, that the pathogen populations can shift fairly quickly in response to selection pressure. So for instance, previously rare pathotypes may increase rapidly if they have a selective advantage in terms of being able to infect a particular type of host.
Okay, how many genes are involved in the first generation and second generation resistance? I think you covered that. Maybe you don't know that, but
yeah, I know for many of the ones carrying first generation appears to be derived from Mendel, appears to be a single major gene resistance, but really, yeah, it's hard to say because much of this material is not really available for public examination.
Is there any link between fungicide resistance in predominant pathotypes?
We haven't really looked at the question, and to be honest, at least in canola in Western Canada, there probably hasn't been much in terms of selection pressure for fungicide resistance because it hasn't really been used as a widespread tool to manage the disease. So we wouldn't expect there to be necessarily fungicide sensitivity in these pathogen populations, but really we haven't looked at it, so it's just kind of guess.
Okay.
Yeah, there's one more question, but
okay.
You can answer that in the chat.
Okay.
Yeah. Okay. Thanks again.
Bye-bye. Thanks.
Yeah, so thank you, Dr. Struckoff. And the next speaker is Dr. Mary Ruth McDonald. She's a professor at the University of Guelph, and she'll talk to us about the fungicide, fumigant, or solarization options for the management of soil-borne plant pathogens. Dr. McDonald, would you share your screen?
Yeah, can you see my screen?
Yes.
Excellent. Yes, thanks, Amy, for that introduction. I'll get right into the topics that I'm going to cover. And let's see if there we go. One of them is clubroot. So thank you to Steve for that great introduction to clubroot on brassica crops. I will talk a bit about some of the fungicide work that we've done, definitely discuss fumigants, solarization, and we have done work on calcium cyanamide, but I'm not going to talk about that in any detail. I can answer some questions at the end if anyone's really interested. And the second disease I'll be discussing is Fusarium wilt of spinach. This work was done by a grad student a few years ago. One of the things he did was evaluate 22 different seed treatments for control of this disease. None of them were any different from the untreated check, so I won't be talking about that anymore, but I will talk about fumigants and some other approaches to control fusarium wilt. So clubroot, we know how important it is on canola. It's also important on vegetable crops. And the work that I'll be talking about was all done at our research site in the Holland Marsh. It's heavily infested, and this work is done on high organic matter soil. So about 70% organic matter, quite a bit different from most of the mineral soils where canola is grown. There is a very high level of inoculum in portions of this site, definitely more than enough to give 100% disease severity when conditions are good for disease development. And in almost all of this work, we used a bioassay with a susceptible cultivar of Shanghai pak choi, assessed 5 or 6 weeks after seeding. So I'll talk about fungicides first. And as Steve mentioned, the fungicides aren't really used on canola. Canola growers can't afford that. But vegetable growers have a wider margin, if you will, profit margin, and have used fungicides at different times. In Canada right now, the only one that's registered or recommended is fluazinam, sold as Allegro. Ranman at one time, cyazofamid, was investigated as a potential for clubroot management. It's another fungicide, Nibogen, developed in Japan, tested in some parts of the world, apparently quite effective, but a number of years ago, we were told that it was never going to be registered in North America. And a few different groups have looked at surfactants as a method to control clubroot. The surfactants mostly target the zoospore stage of the pathogen. Dr. Paul Hildebrand in Nova Scotia did quite a bit of work on this and has published. But some of the surfactants are phytotoxic to the plant, as it notes here. And basically, because they're already widely available, no company is interested in registering them for clubroot management. So let's look at some of the data from a number of years ago on fungicides and clubroot. As you can see, this was field trials done in 2009. We were looking at seeding dates for clubroot development, in addition to the application of cyazofamid that was applied as a drench at seeding. So the green bars are the untreated treatments, and the purple bars are the treated. One of the things you can see is that depending on the seeding date, clubroot severity increases dramatically. This is area under the disease progress curve, but the graph for disease severity looks very much the same. And the asterisk indicates that application of Ranman reduced disease severity compared to the untreated check. And then in August and September, there was so little disease. One, there'd be no need to apply a fungicide if the untreated had such low levels of disease, and there were no significant differences. So this pattern of low clubbing early and late in the growing season is one that we've seen quite often. Actually, Sean Westerveld has done some of this work also. So we see low development at cool temperatures. This is much more relevant for vegetable growers than it is for canola growers. And while cyazofamid was effective in this trial, in other trials, we didn't always see that consistent and definite reduction in disease. So there is some some interest in resistant cultivars, and some of the seed companies have been breeding vegetable brassicas for resistance to clubroot. I hope in this picture you can see the stunted cabbage plants that are susceptible to clubroot compared to the resistant ones that are growing well. So we were wondering if we could apply fungicide to a susceptible cultivar, and if that would give us clubroot control that was equivalent to what we were seeing in these resistant cabbage cultivars. So here's work also done in 2009 and also 2010. These 4 top cultivars are ones that are bred for resistance and advertised as resistant. And then Bronco is the susceptible cultivar, and we want to look at the comparison between Bronco treated with Allegro, again as a drench at transplanting, and the untreated. So in 2009, disease pressure wasn't really high. The resistant cultivars were highly resistant. And no significant difference if we applied the fungicide or didn't, but you might think maybe there's a bit of a trend there. However, the next year, much higher disease pressure, 98% disease in both the treated and the untreated. And again, you can see how impressive the resistance was, the genetic resistance in these other cultivars. So to answer our question, fungicide application did not protect the susceptible cabbage from infection or symptom development. So that leads us to fumigants. And over time, fumigants have developed, and maybe rightly so, a bad reputation. And of course, methyl bromide was the real concern. It was never widely used in Canada, but metam sodium and chloropicrin were and still are. You have to get special training if you're going to apply fumigant. In the last couple of years, Canada's brought in additional regulations basically to protect the general public from exposure. You have to post fields before you apply the fumigant. Everything has to be tarped. There are separation distances that you have to observe between the treated area of the field and buildings. So it gets to be quite complicated to use these. And there's also the concern that fumigants kill all the microorganisms in soil, not just the beneficial ones, or sorry, even the beneficial ones, not just the plant pathogens that we're targeting. And this certainly is a concern. We would like to protect those beneficial microorganisms. As you know, we have done trials on both chloropicrin and metam sodium. Chloropicrin needs specialized application equipment. Nitrogen is used as a propellant. And metam sodium can also be injected into the soil using specialized equipment, or it can be broadcast on top of the soil and then incorporated. And that's what was done in our fusarium spinach trials. So we realize that fumigation isn't going to be feasible for canola growers or for large fields, but there may be applications with vegetable growers or to treat areas of an initial outbreak. Again, this work was done on our naturally infested plots, different concentrations of metam sodium. We did compare the 2 commercial formulations, different concentrations of chloropicrin. Initially, we looked at different types of tarp, but then focused on totally impermeable film, and at one point looked at the duration of tarping. And again, the bioassays were all done with the susceptible pak choi. So the first year we did this, there's a miscalculation about how much TIF was needed. The silvery color is a totally impermeable film. And so some regular tarps had to be used, We did have an untarped check, but we also had a really windy weekend after this was applied, and some of the TIF was blown off the treatment. So these became untreated checks as well. This is what the Shanghai pak choi looks like. Sometimes in the untreated check, the plants are killed early on. That allows that additional weed growth in the untreated pak choi. So our 2015 data, this was grad student Justin Robson, we did have relatively high disease in the untreated check, not super high. A couple of our treatments only had one rep that we could use, as indicated by just the straight line. If we look at the VAPAM rates, it looks like we have a pretty straightforward dose response curve here, where better control at the higher rates. With the chloropicrin, the PIC plus, again, we had one treatment where we only had one rep. Unfortunately, or fortunately, this looked like the most effective treatment, but it was also clear that there was not really a dose response for chloropicrin. But in general, it was clear that most of these treatments were more effective than the untreated check. So we continued this work for a few more years. I won't show the 2018 data because there was very little clubroot that developed in our trials. Again, on our muck soils, these were applied mostly in June, different rates of chloropicrin. We had the uncovered, untreated, and we also had covered untreated. And the tarp was all left on for 2 weeks. I do want to make the comment that applying these in a research situation is a little bit difficult. Sometimes we end up with soil on top. Sometimes you get water puddling on the tarp. There are commercial setups to apply TIF and also the fumigant. And I expect from the pictures I've seen, they do a much better and obvious and more efficient job. You get better bed formation, and you don't end up with soil or other things on top. And of course, if we're looking at solarization, anything that's on top of the tarp is going to reduce the level of solarization and heating that takes place. So we look at our data. Here we have the untreated check covered with totally impermeable film. We see a significant difference from the untreated, untarped check of about 50% and high disease severity, over 80% disease severity. And then when we look at all of our fumigant treatments, they're no better than just the TIF by itself. Maybe a slight trend here, but certainly nothing significantly different. So we're wondering what's happening. Are we seeing a strong effect of solarization with the TIF for 2 weeks? So the untarped, the average temperature was about 22 Celsius. It did increase in the tarped plot, but these temperatures are considerably lower than those published for solarization to control clubroot on mineral soil. So it does make us wonder if we're seeing some anaerobic soil disinfestation activity here. And I'm very much looking forward to the presentation later on today on this topic.
5 minutes left, please.
Thank you. Again, in 2019, we got this same trend. Everything's the same as the TIF. And then when we looked at the duration of tarping, in 1 week, there was no difference, but 2 weeks and 4 weeks seemed to be equivalent. So 2 weeks seemed to be an optimum time. So we did see that fumigants reduced clubroot, but the solarization or anaerobic soil disinfestation with TIF looked to be very effective. And it was important to note that nothing completely reduced symptoms to zero or completely controlled the pathogen. So we wonder if we can use cheaper tarps. Probably not. Looks like TIF is really good. Can we include low rates plus TIF? Might have an advantage, but certainly to use this in practice, timing's going to depend a lot on your soil type and where you are. But I just wanted to note again that without TIF, previous trials had shown that you needed several weeks of solarization, up to 10 weeks to be effective, and temperatures of 40, 45, or 50 Celsius if it's straight solarization. So I just want to talk quickly about some of the work done this time on mineral soil on Fusarium wilt of spinach. Again, spinach is a short season crop, so disease was assessed at 6 weeks. It's bunching spinach, so it's harvested by hand. And that means that if the incidence is higher than 10%, they abandon the whole field because it's too difficult for the hand harvesters to sort out the diseased crop. Can be severely affect the plant, and sometimes vascular discoloration is also assessed as an indication of infection. So we looked at metamsodium, chloropicrin, some other treatments that, like spent mushroom compost, had a non-treated control, of course. The high nitrogen check was the equivalent of 200 grams nitrogen. This is because the muskro, the seed meal, contributes that amount of nitrogen when it's applied to the field. So in 2014, that was the only time that we had chloropicrin. It was very effective. It was tarped, but only for 3 days. Far more effective than any of the other treatments. Very few of them were significantly different from the untreated control. The brassica seed meal numerically had even more disease than the untreated check. And it turned out, just over the 2 years of the trial. Metamsodium did decrease incidence compared to the untreated control, but the really exciting thing about this was that the high nitrogen check was equivalent to metamsodium for incidence and severity, and actually was the only treatment that increased yield over the non-treated control. So to conclude, applying fungicides to control soil-borne diseases is difficult to do well, and it is inconsistent. It's hard to be consistently effective. There are a few exceptions to this. Seed treatments of fungicides for onion smut, that's very effective and actually essential if you're growing onions. Even with fumigants, it's pretty much impossible to eradicate the pathogen from the soil or to end up with 0 symptoms. We find that sometimes fumigants are the only effective option, and people are still, of course, looking for alternatives to fumigants. We're also finding that the carryover effects of fumigants can be very short. Often by the end of the growing season, the pathogen reemerges and lots of the beneficial microorganisms do. There's some data on the return of trichoderma, for instance, following fumigation with chloropicrin. So the solarization or anaerobic soil disinfestation is very interesting now that this totally impermeable film is available. And as I mentioned before, I'm really looking forward to hearing more about that. So I want to end by acknowledging the funding sources, including Canola Council of Canada, Canola, OMA for University of Guelph Partnership. And I think there will be time for a few questions.
Yes, thank you, Dr. Mary Ruth McDonald. Any questions for Dr. McDonald?
Yep, there's questions coming in on the chat. So the first question, fumigation can be very expensive for canola growers if conducted in large acreages, but what do you think about the possibility of fumigation of patches?
This is one of the reasons that we looked at fumigation. I was at a meeting with Canola Council people a number of years ago, and they thought growers might consider this, especially if they had an outbreak of one of the new virulent pathotypes. So of course, the cost-benefit analysis would have to be done. It's definitely possible. One of the things that I found interesting is that there are a few companies in Ontario that do custom fumigation. They don't seem to be very prevalent in Western Canada. So having access to some of these might be difficult, but there is still the option of applying VAPAM as a broadcast and working it in, which is work that Steve Struckoff and Xiaofeng Wang's group has done in Alberta.
Okay, the next question, how do you inoculate plants and control inoculum density?
Well, we do that in the growth room all the time. We grow the seedlings and then apply, we extract inoculum from clubs, quantify it, and apply a known quantity. But in the field, it's difficult, even with our relatively small plot area. We do see differences in the levels of inoculum. So well-established methods for controlled environment. In the field, it's still tough.
Okay, the next question, do you have data on how much are the pathogen populations reduced after fumigation? You mentioned that at the beginning of the season, for example, pea brassicae populations were about 10 to the 6 resting spores per gram of soil. To what levels were they reduced?
Really good question. And even with our 2017 trials, we're still working on that. The molecular methods to quantify resting spores in soil are not as easy as they seem. And when we're extracting from really small quantities, we find a lot of variation. So we will have that data, but we don't have strong data yet.
You're muted, Sean.
Sorry about that. Soil moisture is important for soil fumigation. Have you looked at the effect of soil moisture and fumigants on the disease incidence?
Okay, soil moisture is really important, and there are definite guidelines about how much moisture you need in the soil before you fumigate. So we just made sure that we had enough, we had the appropriate level of soil moisture before we applied the fumigant. And that's going to be crucial for anaerobic soil disinfestation also.
I think we're running out of time for
there's more questions in the chat if you want to answer them, Mary Ruth.
Okay, sure.
Thank you again, Dr. McDonald. And next speaker is Dr. Fouad Dayef from the University of Manitoba. Today, he's going to talk about verticillium wilt in crops, consideration towards better. Dr. Dayef, will you share your screen, please?
Yeah, please give me just a second to share the screen.
It looks good.
Okay, is it the whole screen?
Yes.
Okay, thank you. First of all, I would like to thank the organizing committee for this invitation. I was asked to provide a general overview of on verticillium wilts and their importance in Canada, as well as on management strategies for better management of this disease. And given the time, unfortunately, I will not be able to present results from my work, but rather give an overview of the issue and maybe an opinion on approaches that should be taken forward to deal with verticillium wilts. So I have to stop sharing and sharing again because my slides are not moving. Okay, here we go. So, yeah, I was saying that I would be talking more in general terms and providing an approach for the future, maybe. So crop wilts can be easily confused with other conditions caused by either biotic or abiotic factors. A few examples include root rots, drought and excess water, soil compaction, and other non-infectious disorders. In fact, crop wilts can be caused by different organisms, including bacteria, fungi, viruses, and in some cases, nematodes as well. Even with fungi, for example, sometimes there is confusion as to which fungus is causing these wilts. So, for example, between Fusarium and verticillium in some plants. Here I will be talking only about verticillium wilts caused or wilts caused by verticillium species, which are soil-borne fungal pathogens. The main I don't know why so the main pathogenic species are Verticillium dahliae, which has a wide host range, Verticillium alboatrum, which infects species such as potato alfalfa and a few others, Verticillium longisporum that is associated with the Brassica family, Verticillium alfalfa that infects lucerne, and non-alfalfa that infects hop, potato, petunia, spinach, and probably a few others. Other species include also Verticillium tricorpus and Isaaci, which were reported to be associated with wilts in other crops. But really the most notorious species are Dahliae and Alboatrum, which cause billions of dollars in annual crop losses worldwide. So as I just mentioned, Verticillium can infect a plethora of plants such as vegetables, fruits, field crops, shrubs, and trees. Interestingly, it hasn't been shown to infect any monocotyledons. And the symptoms, as you can see here, for example, on sunflower, they're usually visible on the leaves, starting from the ones at the bottom with chlorosis and then necrosis. Stunting is also a symptom that can be seen with some strains in some crops. And plants also develop internal symptoms that can only be seen after sectioning the roots or the stems, as we can see here on these cross and longitudinal sections of a potato stem to the left. Such vascular discoloration can also be seen on other organs, such as in tubers here. And eventually, as the plant collapses, there is formation of microsclerotia, which are the resting structures that actually allow the pathogen to overwinter and stay in the soil for several years, even in absence of a host plant. When microsclerotia sense the presence of a host, they germinate and the mycelium moves towards the roots to infect them. We're lucky to actually catch this microsclerotia germinating and presenting those verticills. And that's what starts the disease cycle. The infection starts in the roots, and then the fungus colonizes the vascular system. I tried to show what happens here, for example, where the verticillium reaches the roots and then goes through the vascular system towards the top, using the flow of the water and nutrients in the xylem to reach the top of the plant. And it's actually the conidia that do the traveling, because once, as you can see on the second picture, the conidia are larger than the pits or the connections or the apertures that are connecting the different vessels. And once they reach the top of the vessel, they actually germinate, form mycelium that goes through the opening. And once on the other side, it produces more conidia, which continue the flow to the top. And that's how it travels so quickly inside the plant, as opposed to allowing the mycelium to simply grow from bottom to top, which would take way more time. As a result, the plant tries to respond by forming different types of structures, such as tylosis and bubbles, and all imbibed in biochemicals that the plant is producing in an attempt to stop the pathogen from moving forward. But unfortunately, this is what actually makes the plant to have the wilt, because it is stopping, at the same time, it's stopping that flow of water and nutrients to the top of the plant. And eventually, the plant dies and collapses, and microsclerotia are formed and return to the soil and wait for the next host to come. So how does the disease impact economic crops in Canada? Unfortunately, data about the impact of verticillium wilt in Canada are relatively scarce. There is information about potato early dying syndrome in potato, which is another name for actually verticillium wilt with other pathogens involved that may cause out to about 50% yield losses, but I believe Dr. Al-Mughrabi will talk about this later during the day. There are a few reports in the Canadian Plant Disease Survey on other crops affected, such as a number of flowers and strawberry, cucumber, woody plants in BC, tomato, sunflower, and others in Manitoba, eggplant, potato, tomato, cucumber, and other crops in Quebec, and other crops in Prince Edward Island. But apart from a few publications and very occasional surveys, many of these reports are based on visual observation on plants and culturing of the pathogen from samples that are presented to provincial diagnostic services. So that's the first consideration I would talk about, is that there is much need for more consistent surveys for this pathogen in different parts of Canada that would actually be of interest to different crops. So what's the difficulty in managing these wilts caused by verticillium dahlia in particular? Verticillium dahlia has a white host range, as I said, and that counts more than 200 plant species. In some publications, they talk about 400 plant species, and that makes the rotations really difficult to implement because most of the crops that we would like to put in place would probably be infected by verticillium. Verticillium dahlia produces microsclerotia, and that is something that is also present in longisporum that infects canola and other brassicas. And so because of this, the pathogen can survive in the soil for long periods of time. 14 years are mentioned or more, and that makes also the rotations ineffective. So even if you have the right crops, because these microsclerotia can stay in the soil for so long, it is really difficult to implement. The other thing is that verticillium is soil-borne, and that means it enters through the roots, which makes it inaccessible to fungicides. And except, of course, if you do fumigation, but then you kill so many other beneficial fungi and bacteria and other beings. Now, the one important part I think that is really also very important is the biological diversity. The biological diversity of verticillium and its genome plasticity. This pathogen has an aptitude to adapt to conditions and to plant species that is very remarkable, and that feeds its biological diversity, which makes it more difficult to deal with. There are a lot of publications because people have been working on verticillium for a long time, and there have been studies determining races on different crops. For example, in tomatoes, we know that for a long time, we knew there were race 1 and race 2, and now they're talking about race 3. There is also the pathotypes, the defoliating and non-defoliating pathotypes in, for example, in olives and in cotton. And then there is the what's called the vegetative compatibility groups. And this is one tool that really is quite powerful in determining strains that belong to different groups attacking different plants. And there's also, or there have been recently more studies about clonal lineages, but those were not very successful in associating the clonal lineages with strains that would attack one or other plant species in particular. And there are also studies that have combined different tools from these. And I think second consideration is that this is where probably some of the work has to be done with the local or the regional strains that are found on Canadian soils.
4 minutes left, please.
Okay, so I will try to be as fast as I can. So let's talk about integrated disease management practices and how we can do things better. Of course, preventive cultural practices are known to be used in this case, such as the use of healthy seeds, avoiding new inoculum introduction, for example, movement of soiled machinery or soil or people, the use of tolerant varieties. This is an important point because tolerant varieties, when they are developed by breeders, they are only against certain strains, and the difficulty arises when you have new strains that are that don't necessarily belong there. Performing soil tests for Macrosclerotia counts, for example, that has its own difficulties as well, because there is sometimes confusion with other sclerotia from other pathogens. PCR tests, people who do PCR on soil material know how difficult and how inconsistent they could be. But knowledge-based decision-making is where to go. Knowing cropping history, for example, of the field for previous rotations, if any, combined with the knowledge about the population structure, composition, for example, it all depends on the crops we're talking about. It could be definition of the pathotypes or the races or the VCGs or more of those tools. Biological control, cover crops, and green amendments can help. We have done many studies around and about this, and other colleagues have done as much as well. Chemical control can be also integrated into the system. Solarization is something else that people have used a lot in Spain for verticillium. So know your enemy. That's something that has to be done with consistent surveys to establish a map of the verticillium species in Canadian soils used to grow different crops. Analysis of the population structure of these strains that represent a threat to economic crop is very important. This alone will provide a better information for more informed management and decision making. And based on that, we can engineer, I'm not talking genetic engineering, but just establish control measures that are based on that knowledge. For example, establishing studies that will generate useful information for breeders to develop tolerant varieties to the relevant strain. And I'm highlighting that because that's what's important. It has to be to develop towards specific strains for local, regional, or with specificity for some crops. Interestingly, Verticillium dahlia has a white host strand, but we need to know that for each strain alone cannot infect all crops with the same strengths. So we have to make proper choices of crops when knowing what strains we have, because some strains, maybe they are very strong on certain crops, but they are not on other crops. We have to support integrated management strategies with other biological methods, such as biocontrol. And there is plenty of studies that have shown the efficacy of some of those using plowing, cover crops, and biofumigants also have been published and have some interesting perspective there. So we have published a lot on verticillium in the last few years, but we have looked at host pathogen interactions, looking at effectors genes from verticillium that allows it to do a good job on certain crops, some genetic and proteomic and other studies to differentiate different verticillium strains from different crops. But as I said, it has to be a collective effort to focus on the consistency of the verticillium that we have in different soils in Canada to do a better job in the future. So I hope I did not take more than the time I was, for some reason, my system did not work. I had to do everything manually here. That's what Zoom does to you. And finally, I just would like to acknowledge all the people who contributed to my work on verticillium, from students, technicians, research associates, and different collaborators here in Canada and elsewhere, as well as the funding from NSERC, RD, MRAC, MAFRE, McCain Foods, Dow AgroSciences, National Sunflower Association, and Keystone Vegetable Producers. Thank you very much. I would be glad to entertain some questions if we still have time.
Thank you. We have a time for one quick question for Dr. Dayev.
One question in the chat: can verticillium enter fruit, example tomatoes, and spread the pathogen via culled fruit in healthy fields?
Well, verticillium certainly, well, it enters through the roots, and it travels through the vessels to the upper parts of the plant. I don't remember. I remember seeing infections of tomatoes when you open them, having mycelia of, but I don't remember if it was Fusarium or verticillium. But honestly, I don't know.
Okay, thank you. If you have more questions, you can just put your question in the chat. So I'll welcome the next speaker, Dr. Bruce Gossen. He is the a research scientist at the Agriculture and Ag Food Canada in Saskatchewan. He'll talk about Razoctonia in field crops, biology, and issues with inoculum density. Dr. Gossen, if you can share your screen, please.
I'm trying.
Fuad will have to stop sharing.
There we are.
Okay, Bruce, you should be able.
Okay. Again, like everyone else, thanks to the all the other speakers, thanks to the organizing committee for inviting me to do a presentation this morning. I was invited to talk about Razoctonia, and so we're going to do a very short and quick review of the biology and pathology of Razoctonia on a variety of crops. But one of the things I really wanted to talk about was issues with inoculum. And so once we've done that quick review, we're going to come back to issues with inoculum and hopefully have a chance to deal with some of those in a bit of detail. Okay, Razoctonia. The Razoctonia is a genus in the Basidiomycetes. They have teleomorphs, but the teleomorphs aren't important in etiology or epidemiology. Razoctonia solani is the most important crop in this, or pathogen in this group, but there are certainly other important pathogens. The picture in the bottom right of purple carrots are infected with another Razoctonia species, and that's just to remind me that there are important pathogens outside of this group. But for field crops, Razoctonia solani is the one. Raza is Greek for root, and tonis is for murder, so it's a root killer. And it was originally described on potatoes, so the species name comes from solanum, which is the name for lightshade, or nightshade rather. There are 13 anastomosis groups. We don't have very much. There are no really spores to look at, so, and very little from the mycelium that is distinctive. So we use anastomosis groups to identify the type of Razoctonia solani that's equivalent to the vegetative compatibility groups that Fouad was just talking about. In addition to the 13 anastomosis groups, there's, or AG groups, I've used that throughout the presentation. There's one binucleate group and often subdivisions within each AG group. They're almost certainly polygenic in that there are multiple species that are included in this disease complex species, but we still have trouble differentiating them, even using molecular approaches like sequencing of the ITS region. And of course, the pathogen is endemic across the temperate regions of the world.
Sorry to interrupt. You're on the presenter view. Do you want to switch the regular presentation view, or are you okay with that? Because people can see your next slide and the notes section.
Oh, I'm sorry, I can't see that, but
yeah, if you go up there, the displayed settings on the top of the I don't know if you're seeing the same way as
display settings.
Yeah,
oh.
And then click that, and then swap presenter view and slideshow. Yeah, we're good now.
Thank you very kindly. Okay. So you all know the characteristics of Razoctonia. It's one of the first ones that we ever see in a first year pathology class. So I'll just move on to the next slide from there. Razoctonia causes mainly damping off and seedling blight, resulting in weak stands of plants and often early plant death. But it also causes root rots and even foliar disease, and often as part of a disease complex. There's Razoctonia solani has a very wide host range that includes basically all of the major crops in Canada. But the interesting thing is many of the isolates are at least partially host specific. Some AG groups have a wider host range than others. And we're going to come back to and talk about that a lot more. In terms of management, I'm not aware of any strong sources of resistance to Razoctonia root rot in really any crop. That I've not done a comprehensive survey, but at least for field crops, no good sources of resistance. And just like the verticillium that we were just talking about before, cropping rotation has very limited applicability here. We know that in wheat that short rotations increase the population of specific AGs that are aggressive on wheat, and we assume that that same relationship occurs on most other crops. Even though because of the difficulty of identifying AGs and the time-consuming aspects of it, that hasn't been done for every crop, every AG on every crop. One of the effective approaches is fungicide seed treatments, and they protect seedlings from damping off and often seedling blight, but as you might imagine, they wear off quickly and don't protect older plants from root rot. There's lots of papers about biological control agents, but as far as I know, there are few or none that are being used on a commercial basis, and certainly not on field crops. As with many other kinds of root soil-borne pathogens, limiting movement of infected soil is a really good way to limit the movement of and infection of individual crops and fields. However, it may be too late for that to be a useful strategy in most agricultural soils, because the pathogens and AG groups are ubiquitous. Okay, why isn't it moving? Okay, in terms of the impact of the pathogen, it causes damping off in canola, pulses, cereals, and soybean, and seed treated with fungicides is planted on literally millions of hectares each year in Canada. That's a substantial application of fungicide. It's a substantial expense to growers, and so clearly is very important. The pathogen itself is often considered a weak root rot pathogen in mature plants or older plants, but I've included a picture here of Razok on potato to show that it can be an important root rot pathogen in certain situations on certain crops. In terms of assessing the impact, there's an important issue, and that is it's so difficult, or we don't know how to measure pathogen load, disease pressure, pathogen distribution, and we'd like to be able to quantify that using various molecular techniques or a molecular technique, but right now those quantifications are difficult or not possible at all. This is a similar issue with lots of other root rot pathogens, but in addition to the standard problems with other root rots, we have not spores or oospores or defined units of inoculum in soil, but we use what we call colony forming units, which is units that are big enough to grow on agar on selective media. And we also have the problem with these anastomosis groups that can't be distinguished one from another. Now, this is a picture or a slide taken from a paper on clubroot, but the same kinds of issues are present with Razoctonia, that the particles that are involved in causing disease are actually small enough to move easily in wind and water, and also to be carried on equipment. Probably for most situations, equipment is the most important the mechanism for moving material from field to field because of the volume of soil that's carried. Here's an example, again, not on Razoctonia, but this is clippered on canola to give an idea of another important mechanism of movement of soil-borne pathogens. Here, a small infection in a low area of the field was almost certainly carried up from the low area into the rest of the field with a cultivator. That straight line, those kinds of straight lines don't occur in nature unless we help. I'm sure that this grower moved that material not only into the area where the plants are killed, but throughout the field by carrying disease materials on the cultivator shafts. Another very important problem in working with soil-borne pathogens is the patchy, non-uniform incidence or distribution in fields. Now, this is a heat map of a research trial that was done at the University of Guelph back in 2015. Mary Ruth McDonald actually showed you pictures of this trial, but the area that I've put in the box there shows a portion of the field where disease pressure was very low, actually too low to be useful to us to study, even though we chose that area very carefully to try and provide a uniform disease. So the light green here is very low disease, and the red is high disease. So with to overcome the problems with non-uniform, patchy inoculum distribution and disease pressure in fields, researchers inoculate their trials often, and this is an approach that Kanfa Chang, Shofang Wang, and I used in numerous studies that were done back in the 2000s and or actually 1990s and 2000s with the Razoctonia on the prairies on pulses. And in order to do that, what the process is first to select a site that has little or no inoculum, and you want to do that to eliminate problems with background levels of inoculum. But there's a question that always comes up with that: are such or are sites that have little or no disease actually conducive for the pathogen? So that's a question. Is the pH right? Or the soil texture right? Is the moisture conditions appropriate? The second thing that you do in those kinds of trials is you select a highly aggressive isolate. And again, you want a highly aggressive isolate to give you consistent disease. But as a question, does an aggressive isolate actually indicate the real disease potential in the field or the region? Or are you really picking out the 1 in 1,000 isolate that is going to cause a lot of damage? And lastly, you select an inoculum rate that's high enough so that the disease develops consistently, irrespective of what the weather does. And again, how is that relevant? How relevant is that kind of rate to an actual field situation? Now this is from a published study on lentil, Razoctonia on lentil, and in order to get an estimate of how much inoculum we should provide, we assessed two lentil cultivars with root rot severity on the left and a log of inoculum density on the right. And you can see that with that log inoculum density, there's a straight line, which means it's it's actually an exponential disease curve. And it very much depends, how much inoculum is enough, very much depends on how much disease you are trying for. So you need 10 to the 2 colony forming units per gram of soil. If you want to get high levels of disease, you need probably only 0.2 colony forming units per gram if you're willing to accept a much lower level of disease. Somewhere in between those two may be the amount that gives you the levels of disease that are actually common in fields in the region.
5 minutes left, please.
Okay, thank you. However, there's a problem with too much inoculum, and that is that you can easily overwhelm the level of your treatment with even a highly effective treatment with too much inoculum. I'll just keep on moving here. Here's an example of that, again from clubroot rather than Razoc, where we were looking at the effective pH on infection at 3 different inoculum levels. At the low level, we got a very strong reaction to pH. At an intermediate level of inoculum, just a barely measurable response, and at a high level of inoculum, no response whatsoever. So we know that there are problems with inoculation, even though they can obscure treatment effects. We also don't know about colony forming units and how that relates. And also there are many isolates that are not aggressive, but there's multiple anastomosis groups often infect a single host. And then there are all kinds of problems with molecular quantification, the most important being how, when, and where to sample. So with Razoctonia, it strikes me that it's possible that we're using too much fungicide. At present, almost every seed that's planted on the Canadian prairies is treated with fungicide because it's considered cheap insurance against seedling blight. But some recent meta-analysis on soybean in the US shows that many growers would actually would have more money in their pockets without a seed treatment. And I wonder if we have oversold the importance of seed treatments because we're using high rates of inoculum in the studies. There's one other little thing that I want to talk about very, very quickly, and that's balancing selection. In what we call balancing selection, is a situation where entire genotypes are retained in the pathogen population over time. We've seen that in cliproot, where the seedling becomes infected with usually actually potentially hundreds of separate infections occurring all at the same time on a young seedling. And we know that the aggressive pathotypes will suppress the disease reaction or the disease response of the host, and that allows not less aggressive or even non-virulent isolates to get in and create at a low rate and develop spores and maintain the population, maintain their presence in the population. And that explains how that particular pathogen can so quickly overcome resistance, because there are lots of different genotypes available. In Razoctonia, we're seeing a situation where there are multiple AGs that are commonly found from infected roots, but most of them aren't really very pathogenic. How and when do they get into the infected root? And is it possible that in fact, I put forward as a hypothesis that once an aggressive AG infects the root, it suppresses host resistance, and others could follow it in either as weak pathogens or as actual saprophytes, and so that all the AGs increase when one increases. If that's the case, balancing this would provide for balancing selection and maintenance of the population as a whole. So in terms of steps forward, I think we need to do multi-site trials in commercial fields to confirm the need for routine seed treatment. And we need to improve our sampling protocols to identify, to stratify them based on plant stress, and perhaps aerial photography would help with that. We need molecular approaches to identify specific AG groups, and we need to use those to quantify AGs from the soil. We need to study the changes in these AG groups over time using those molecular approaches. We need to develop molecular techniques like digital drop PCR to define what a colony forming unit is, to help our maintain our understanding and to connect the current work that will be molecular with older studies. And I'd love to see some work to demonstrate or disprove the hypothesis that balancing selection is occurring in this group. Now, there's been lots of funders that have provided funding for the work that underlies this presentation. And I'd really hope that there's time for at least one or two questions if I can answer them. Thank you very much for your attention.
Thank you, Dr. Gossen. Is there any questions?
I have a question. So ginseng growers, they deal with Razoctonia is quite an issue for them, but they have been able to control it very effectively with azoxystrobin application at seeding and in the following spring. But they're not using a seed treatment; they're using a fungicide. They apply it before seeding, and then it basically washes in with the rainfall. Do you think that kind of approach would potentially increase the risk of resistance developing in the pathogen? Because when it's a seed treatment, you're only influencing what's directly near the seed, but when you're basically treating the whole soil, you're impacting everything in the soil.
Oh, that's an interesting question. Certainly it's increasing the exposure of non-targets, and so other kinds of organisms or other crops following may have more issues with fungicide insensitivity. But I think the things like azoxystrobin bind to soil so quickly that the long-term exposure to these organisms is not likely to be an issue. So I think that there's a possibility, but I don't see it as a high probability. Does that make sense?
Yeah, that makes sense. I mean, they've used it for quite a few years, and I haven't seen any issues. But so there's just a comment in the chat. Balancing selection is an awful phenomenon. I think we should make this type of evolutionary process illegal.
I'm all for that, but I don't think that the pathogens are so very concerned about being sued.
Yeah.
Thanks, Clint, for your comment.
Yeah, I think we're running out of time for
questions.
questions.
Marlene, just one quick question. Marlene?
Yep.
Yes, Dr. Gossen. I run the plant disease diagnostic lab for PEI, and each year I do receive a lot of potato samples. And I have to say, I think a lot of our inoculum we have here is from their seed, is from the potato seed. I suspect there's a high level in the soils here as well, but nobody's really looking at the seed sometimes because they don't tend to wash the seed, and the sclerotia is always on the potato tubers. And then when I get samples coming in, I see it both on the stems. The lesions are on the stems, and oftentimes I'll have a conglomeration of organisms coming from the stems as well. So I guess my question to you is, like, if you have Razoctonia lesions on the stems, is it always coming from the seed and or the soil with potatoes?
What would you say? Is it always coming from the seed or the or the soil? I would say yes. Yes, yes, both. The pathogen is endemic in a lot of places, but I think one of the places, like a lot, but when you do the assessments in soil, you're going to find that a lot of the materials that you isolate aren't going to be pathogenic on potato, even though they increase over time. And so that's why we need better tools to identify what we've got, so we can separate out the ones that are actually likely to cause disease from the ones that are just hanging around and being carried forward.
Good point.
Okay, thanks very much.
Yes, thank you.
Thank you, Dr. Gossen, for a great presentation. So we'll move on to the last presentation before the break from Dr. Tahara Satana from the Agriculture and Agri-Foods of Canada. She'll talk about the nematode involvement in fruit tree decline disease in Canada. So if Tahara, if you can share your screen, please. You're on the presenter view. If you go to display setting and then change that to I know we can't hear you.
Tahara, you can do that within Zoom.
So you can just share, start your presentation, start a slideshow. Oh. Are you
Sorry,
able to, sorry.
We can't hear you. You can try dial, if you can't get the speak sound to work, you can dial in to the number on the thing through the phone.
Sean, could you unmute her, please? See if that works.
No, I can't. I have to ask to unmute.
Oh.
Still can't hear you. No, no sound yet. We can see you, Tahara. So if you have a phone nearby, you can call the number on the invitation. I can put that in the chat if you want.
So are you guys able to see my presentation?
Not in the slideshow mode yet.
better now?
It's in the presenter view. If you go on the top, display setting, there's a little triangle. If you click that and then do swap presenter. Yeah, here we go. We're good now.
Perfect. Thank you, and sorry again, and thank you so much for your patience. Thanks to the organizer to giving me the opportunity to present my work in front of you. Today I'm going to talk about nematode involvement in tree fruit decline disease. It all started when our local growers start to see the sudden death in their orchards. One of them was sudden apple decline, which called Asd. This is a disease for their apple orchards, and another is fruity decline, which is a disease for their tree fruit orchards, which is the palm, peach, apricot, etc. So for the Asd, the mortality rate is up to 40%. Most of the newly planted trees are dies off, and this collapse is very sudden. It could happen within 2 weeks. And on the other hand, for the fruity decline, the mortality rate is higher than Asd, which is 60%, and same as Asd, newly planted trees dies off within 2 years. So if we're gonna see about the common symptoms, what we have for this disease. So there is nothing much actually. All in the sudden, we can see that branches are starting showing smaller or fewer leaves. Tree collapse all in a sudden. You can see a chlorosis happening or wilting of the tree. Some of the cranker could shown on the trunk, and all in a sudden the whole plant is dead. So this is actually all we know about this disease. So if we try to understand what is this is completely unexplained death. So you have a complete healthy orchard, and all in a sudden you see that branches are starting to die, and without any reason or without any sudden symptoms, you see that your plants are dead. So these symptoms actually vary from orchard to orchard. We actually don't know actually what is the exact symptoms of this disease. And also, obviously, we don't know what is the cause of this disease. So a group of scientists started to investigate on that, what might cause this disease, and thereby a complete action plan has been made. So our research approach started with identifying the disease orchards and choose sampling sites across Canada. We included British Columbia, Ontario, Quebec, Nova Scotia, and Prince Edward Island. We isolate and characterize pathogens associated with these two Asd and STD disease. We determine the involvement to known and new organisms associated with this disease. We want to identify the pathogenicity of these different organisms in Canadian orchards. And finally, our aim to examine the effect of combined stress on tree fruit death. So as a nematologist, my interest obviously goes to see and understand the involvement of nematode with these two important diseases that our local growers are facing right now. To go on that purpose, several samples have been collected from different affected orchards around us. So in terms of apple, we have collected 29 samples from 4 counties of Southern Ontario, where most of the apple orchards are located. We also have 33 samples from 5 counties for apricot orchards. We have 8 samples from 2 counties from plum. We have 17 samples from 2 counties from peach orchards. Total 26 samples has been collected from 1 nectarine, and total 7 samples has been collected from 1 sweet cherry orchard. So sample processing has been done as a regular way, how we do in our nematode lab. We did sampling in a zigzag style. Nematode was extracted from 50 grams of soil using the sugar flotation protocol, and plant parasitic nematodes were identified at the genus level by using inverted microscope with several morphological characteristics. With the deep investigation with the 37 samples that we have collected from apple orchards, 27% of them are positive to lesion, 10% of them are positive with ring, 75% are positive with dagger, 34 with pin, and 41 with spiral. In terms of average population density, the highest population density we found for the dagger, which is 482. Other than that, we have 34 for lesion, 4 for ring, 57 for pin, and 73 for spiral. If you see this graph here under, you can easily see the high infestation rate for this dagger nematode, which is way over all the other nematodes we have found here. Also, in terms of nematode involvement in the fruit tree decline disease, we have collected samples from apricot, plum, peach, nectarine, and sweet cherry. Most of the samples were collected from May to July in 2019. In between the 33 samples that collected for apricots, we have 63% orchards positive for lichen with an average population density of 123. We also have 51% orchards positive for ring with an average population density 206, 45 positive for dagger with a density 82. The highest came for the pin, which is 69 with an average density of 468. Similar for stand and spiral, we have 12 and 15% orchards positive for these two nematodes, and average population density is 18 to 25. We actually did not test it other than these 6 species because those are the most common species found in our local soil. In terms of plum, we have the highest density goes for ring, which is 100% orchards are positive for that, with a very high average population density with 1190. And second goes to dagger, 75% with an average population density of 375. We also have all of the nematodes present in the same orchard. In terms of peach, we have the 52% positive for lichen with an average density 70%, 23% positive for ring with an average density of 18. But the highest goes for 94 for both pin and stand with an average density of 1710 and 267. We have only one orchard for both nectarine and sweet cherry. So this is actually not obviously representing all the orchards around us, but however, we also have presence almost all the nematodes in here. So as you can see, most of the numbers is associated with lichen, ring, dagger, and pin. So I took the study in the second level to see their population density, how much they're actually present in the soil, and what rates of infestation happening. So for the apple, we have the highest infestation rate with the Diffinema, which is dagger, that causes more than 75%. And the number of this 75% is way more than the threshold level, which is more than 100 per kilo of soil. We also have presence lichen, ring, and pin nematodes. In terms of apricot, similar situation is happening here. We have more than 75% of the orchards is highly infested with ring nematodes. More than 25% is highly infested with dagger nematodes, and almost 50% is highly infested with pin nematodes. In terms of plum orchards, we have almost 80% orchards highly infested with ring, and 100% orchards are highly infested with dagger nematodes. In terms of peach, we have 25% of the orchards which have very high number of ring presence. We have about 50% of the orchards which has very high number of dagger presence, and we have more than 75% of the orchards with a very high number of pin nematodes present. We do not find any lichen nematode for our nectarine orchard. As I said, that this is only one orchard we have collected samples from. That was our ASC Jordan farm. But however, still we have a lot of Diffinema and Sartulenchus present in our samples. Similar to our sweet cherry, we have only one site sample. If you can see, the number of ring infestation is very high in here too. So from all my previous charts with apple, plum, peach, nectarine, sweet cherry, or apricot, you can see that the color red, that always comes for ring and dagger. So we try to take this experiment to the next level to see how these two nematodes pathogenicity is happening or really associated with this disease. So on that aim, we have set up a micropod in our ASC Jordan farm. So a split pot design has been maintained and all the plots fumigated before the plantation. The main goal was for organizing these microplots is that we want to see the study or relationship between two rootstocks and two varieties. So we choose the rootstocks and varieties that most of the growers are using around us, which is Harrowblush and Harleen. And also we choose the most common rootstocks that our growers are using, which is K1 rootstock and K86 rootstock. And another purpose for the nematode aspect, we want to see the pathogenicity test of nematode complex. So we have set up a study in 3 ways. Control nematode complex 1, which is the ring nematode only or trichonema, and nematode complex 2, which is trichonema and diffinema or ring and dagger together. So after a lot of hard work of our farm crew, thank you to them, we have set up a complete microplots in our farm. Our farm was fumigated, as I mentioned before, and nematode was inoculated after fumigation and at time of plantation. And this is the farm picture from 2019 that we have, and we also have a very good healthy microplot in 2020. So if we're going to do a deep investigation of our results that we are having from our microplots, we started with our rootstock analysis. So the data from the 2020 that we have, the rate of dead plants is showing that 100% of the trees from the Harrowblush or K1 are dead. 90% of the trees from Harleen on the K1 are also dead. On the other hand, only 10% tree from the both variety on rootstock 86 are dead. So we came to a hypothesis that rootstock K1 is more susceptible to the fruit tree decline disease. However, in 2021, we have replanted all the dead trees to see the long-term effect on that. So if we see the growth comparison for the height of all the microplots plants that we have, you can see the blue line, that is the control. The growth is slightly higher from the other 2 nematode complex, which is 1 and 2. And in terms of diameter, however, we didn't find very significant difference, but this is only the 2nd year of the microplot, so we are hoping to have more results in soon. We have 4 sets of data for all our sampling protocols from our microplot. So for the future research, this microplot will keep in there, and we will have a lot of study data coming for plant growth, leaf chlorophyll, pruning, fruit body, stomatal conductance, root growth, root damage, or metabolism. However, I will keep those data for my next presentation because all data is not ready for my talk yet. We also have a plan to set up a greenhouse study to see the combined effect of nematode and fungal pathogens that my fellow scientists already identified from their research. And also we want to take this study to the next level to see in greenhouse effect and also in farm effect to see the pathogenicity test to understand the effect of abiotic factor and nematode factors together. So I think I talked too fast because I took time in the beginning. So I'm almost end of my slide, which is good for you. You guys can have early lunch. I also thanks an acknowledgement to all my Vineland staff, especially Rio Brandiel, for helping me with the sampling and analysis all those results, and a lot of OMASA personnel to identifying the sites and helping me with the sampling, and especially to the growers to allow me to their sites and having the samples. And this project is funded by our internal ABS fund, Agriculture Agri-Food Canada. And thereby, I thank you to all. I'll have any questions. Thank you to listening to me. And sorry again for the initial technical difficulties. You can send me also questions on email if you want to. That's all for today, and have a good lunch.
Thank you. Is there any question for Dr. Sultana?
Yes, we have in the chat. Were the controls on the K1 rootstock dying as well as the nematode plots?
Give me time, I will just go to that.
You can see it in the
When the controls on K1 rootstocks dying as well as the nematodes plot. For one, yes. For the other one, the 100%, yeah. Also, the control has died. So if you can see here, yeah, the 100% of the K1 for Harrowbush is dead, but the control for the Harleen or K1, however, it is dead.
Another question, were the microplots inoculated? If so, how?
Yes, the microplot all was inoculated. So how we did it, before starting the microplot, we fumigated the plant as the protocol. And then we put the sterile soil inside the microplots after set up the microplots. And then we give enough time to set up the sterile soil in the plots. And then we actually hand-picked all our nematodes to the threshold level and then inoculate them at time of plantation. So this is how we are inoculating our nematodes.
And I have a question. Have you looked at the interaction between fungi and nematodes at all in your study?
So that's our plan is happening in the greenhouse study right now. So my fellow researchers, he already identified 2 fungal species. I don't want to mention the name to take out his thrill to present his result to you all. But yes, that's the plan. So we have ordered the plant maybe in the spring.
Are you doing nematode identification at the genus level? Any confirmation with molecular tools?
So primarily, yes, with genus level. So when we have the samples from the orchards, excuse me, when we have the samples from the orchards, we do the identification under microscope, which is genus level. But for the species level identification, we took it further with the molecular tools, and that's not for all the sites, but for few sites.
Okay, could you explain how you took your soil samples for nematode testing? How many plugs and what amount of soil?
So you mean from the orchards, right?
I believe so, yes.
Yeah, so we identify the total orchards in 3 levels. So healthy plants, diseased plants, and dead plants. And then we took sample around the trees in a zigzag way. Excuse me. And also, we are having the whole field sample in a zigzag way as all together. So then we separate the 3 levels, and then we do the identification from there.
Okay, and one more question: Are you rearing nematode species or isolating from soil for inoculations?
Both. So for ring nematode, I successfully reared them, but for the dagger nematode, I failed to rear them in lab. So for the dagger, we are actually still relying on growers' field. We have the samples and we are hand picking them. But for the ring nematode, we have our pure culture in our lab.
And then one more, are you sampling from 20 centimeters away from the trunk?
It starts from the near, so it's a star way. So one sample 20 centimeter, another sample 50 centimeter, another 20, another 50. And you have like total 10 around one tree, and then you mix them all together and then start your extraction from there.
Okay. I I think that's it for the questions.
Okay, then, and still, yeah.
So go ahead.
So still, if you have any question, feel free to email me.
All right, thank you. And then thank you all the speakers for the first section. We will take about 17 minutes break and I'll come back at 1:35 p.m. Eastern time. And then we have 4 more presentations. Thank you. See you later. Well,
So I should
Dr.
discuss.
Yes, please, if you can stop sharing your screen.
So good afternoon, everyone. We are getting ready to start our evening session. Like it depends on where you are, but our afternoon session, I'd say. First of all, we have our keynote speaker, who's Yoji Muramoto. Dr. Yoji Muramoto is an assistant cooperative extension organic production specialist at the University of California, Agriculture and Natural Resources. He will be presenting today about anaerobic soil disinfestation for soil-borne pest management, defects, mechanism, practices, and limitations. So you're welcome to share your screen. Thank you so much.
All right, can you see my slides?
Yes, we can.
Okay, great. Well, good afternoon, everyone. My name is Yoji Muramoto. I'm a soil scientist, agroecologist, and University of California Cooperative Extension Organic Production Specialist based at the University of California, Santa Cruz. I'm honored and humbled to be invited to this important workshop for Canadian plant pathologists, given the fact that I'm not even a plant pathologist. But I'm very pleased to share my experiences on anaerobic soil disinfestation, in short ASD, a biological approach to control soil-borne diseases with you all today. I thank Dr. Sean Westerveld and the organizing committee for inviting me to this workshop. First, I'd like to thank many people, especially Professor Carol Shannon, the PI for this entire ASD project for the almost last 20 years, and my colleagues and mentor of plant pathologist, Steve Koike, Mark Matora, Erin Roscoff, and many other collaborators, growers, extension specialists, industry people, and the Shannon Lab staff, students, and volunteers who have made this work possible. Also, I'd like to thank funding agencies, USDA, Organic Farming Research Foundation, and California Strawberry Commission support. Those supports are highly appreciated. I'd like to start my talk with a photo of 6 strawberry plants. This picture shows why we started the ASD study in California in 2002. It shows the outbreak of Vertium wilt caused by Vertium dariae at the UCSC organic farm in 2002. The field was under 7-year rotation for strawberry. So once planted strawberries, 6 years of vegetables and something else, and come back to strawberry. Regardless, we have this outbreak. The Vertium wilt, the previous talk, just talk about it, but can be hosted more than 300 species of crops, including important crops in our region, such as lettuce, tomato, potatoes, and cucumbers. And given the small-scale diversified organic farms here, in which we grow more than 40 crops annually, it is hard to avoid those host crops during the rotation cycle. So we ended up have this kind of outbreak for strawberries. Strawberries is one of the most sensitive crop for Vertium wilt. On top of that, in our area, Santa Cruz, we cannot do sterilization As you see here, the summer temperature in our region is colder than Toronto. I'm not talking about the winter at this point, but the summer temperature is really cold and not enough solar radiation to do sterilization. So, we cannot fumigate given the organic farm, and you cannot sterilization. So we're kind of stuck. Fortunately, Dr. Ariana van Bruggen, at that point, she was at the Wageningen University, a friend of Professor Carol Shannon at the UC Santa Cruz, came over to the farm and encouraged us to try newly developed practice called biological soil disinfectation, BSD. And later we learned the same principle approach was developed in Japan almost at the same time independently. So we started to learn from both approaches and trying to optimize to California strawberries. It took us a while, a couple of years to figure out how to do it. But at the end, it controlled Vertium dahlia in strawberries very well. So then, if it works for organic systems, why don't we try as an alternative for fumigant in conventional system? So we started to study as an alternative for fumigant for California strawberries in 2007, and then conducted multiple on-farm replicated and demonstration trials across coastal California. And thanks to that result, and then we got very promising result, and then commercial production scale adoption began in 2011. Now I'm going to show you the current status a little bit later. And commercial organic strawberry nursery adoption began in 2017. And not only strawberries, we tested it for crab root suppression in Brussels sprouts, and it works in the research field trial. And then almond and walnut orchards and nurseries started to try this approach as well. And its research is in progress now. For the rest of the time, I'm going to talk about first principle of ASD and effects, target pests and pathogens, mechanisms, practices, limitations, and next steps. Okay, let's talk about the principle first. The principle of ASD is acid fermentation in anaerobic soil. Basically, it integrates the principle of sterilization and flooding for places where either practice is not effective or feasible. For example, Santa Cruz in our area, California coast, is a perfect example. We don't have enough radiation for sterilization, and the water is so expensive, we cannot do flooding at all. But ASD might be possible. I know Dr. Westville been trying ASD for ginseng, and I know another strawberry grower in Canada has been trying ASD. So there's a potential to do ASD in Canada as well. There are 4-step processes as to how to do ASD. First one is to incorporate locally available labile carbon source. And this is to feed native microbes. You don't need to add any microbes to the soil, but we use the native indigenous microbes, feed them. And then quickly cover with a plastic film to limit the gas exchange and oxygen supply, and moisten the amended soil under the top. Fill pore space with water and initiate the fermentation process. And then maintain anaerobic decomposition fermentation process. And the water doesn't have to be saturated all the time. Maintain above field capacity is fine. And then biological, chemical, and physical changes suppress pathogens and pests. How about the effect of ASD? This table was summarized by a Dutch pathologist. And there they tested a wide range of pathogen for the efficacy of ASD. And fungal pathogens, bacteria, nematodes, and even weeds. As you see there, different pathogen has a different efficacy by ASD. So susceptibility to ASD varies depend on the pathogen. That's an important fact. And for weeds in California, it seems like it doesn't work well. The higher the temperature, the better the effect on weeds by ASD. This table was created by a Japanese pathologist. They originally used wheat bran as a carbon source, but recently they come up with different options, diluted ethanol, 1 to 2%, very, or even lower, 0.5 here. Very thin ethanol. So ethanol itself is not the disinfection agent, just used as a carbon source. And also the advantage of using liquid carbon source is the ease of handling. Also, you can treat the deep soil layer, subsoil, even because carbon source goes down. So some nematodes and pathogen that reside in the subsoil can be treated using liquid form. Mainly horticultural crops is being treated, and they tested clubroot disease by this approach as well. How about in the United States? California, we use rice bran from Northern California as a carbon source. And we've been tested strawberry, Brussels sprouts, walnut, almond. And then we demonstrated that effective for those pathogens. And in Florida, my collaborator seems like they've been trying different carbon sources, but best combination so far is molasses from sugarcane industry over there, and then composite broiler litter. And this broiler litter is not the carbon source, so to speak, but to improve the water holding capacity of sandy soil in Florida. They only have bad sand and good sand, right? Everything seems to be sandy. So that's very important. And the vegetables and cut flowers and strawberries have been tested. And then given the high temperature, some other pathogens can be controlled. Tennessee tested plant residue and molasses, wheat bran for vegetables and sclerotium. And the Washington State, they tested for apple and for replant disease complex. And in Florida, they tested for orange trees as well, citrus trees as well. So how does it work? ASD has multiple mechanisms and working together. First one is shift in microbial communities to create competition or antagonism that suppress pathogens, and the production of organic acid and volatiles. That's very important. And the reduction of iron and manganese, production of Fe2+ and manganese 2+ that are toxic to some pathogens, and lower pH and lack of oxygen itself, and combination of all of these. But the question is, how are each of these processes related to suppression of specific pathogen? And how are processes affected by carbon source used, soil moisture and temperature, and initial microbial community? Those are the questions we're still trying to figure out one by one. This chart shows the management triangle. Carol Shannon put it up together. So as you see here, temperature and water and carbon source type and rate affect the depth and time, depth, intensity, and length of anaerobic conditions. And then under anaerobic conditions, as I explained already, organic acid and volatiles are produced, and microbial community shift takes place, and manganese 2+ and iron 2+ are produced, and those combined and together attack pathogens. And this excellent study, if you're interested in ASD, I highly encourage to read this paper. But this study monitored how metabolomes and microbiome changes during ASD process. And study with aerobic and facultatively anaerobic and then anaerobic phase. And this schematic figure shows how this changes took place. Oxygen and sugar content decreased in the beginning, and then CO2 and the fumigants increased and the bacillus increased. and lactic acid increase here. And then gradually organic acid produce other organic acids like acetic and butyric. And then Clostridium increased the later strong anaerobic phases. And sulfides, highly toxic to some pathogens, are produced towards the end. This kind of shift, microbial community shift and volatile production shift taking place during the ASD process. It's very complex, but those are now we started to understand. To control pathogen using ASD, both temperature and level of anaerobic conditions are important. This just shows the example for Vercium diarrhea control by ASD. And the x-axis shows the cumulative EH millivolt hours below 200 millivolt. Basically means the intensity of anaerobic condition during a certain period. So the higher the number, stronger the anaerobic condition. And then y-axis shows the Vercium diarrhea microsclerotia number in soil. So as you see there, we tried this experiment in 2 different temperatures. At 25 degrees Celsius, microsclerotia number decreased when a cumulative EH millivolt hours exceeded 50,000 millivolt hours. But that didn't happen at the lower temperature. So not just intensity of anaerobic condition, but temperature is also important. Such environmental threshold, we call it, is so important to assure efficacy of ASD to certain pathogens. And so far, we have developed one for Vercium diarrhea, as I showed you before, previous slide. And also for Fusarium oxysporum fragaria, another lethal pathogen for strawberries. Recent study by Henry found much stronger anaerobic condition is necessary to control this pathogen under ASD. And I found, our team found the temperature has to be also higher in the field study. And another lethal pathogen for strawberries, Macrophylla facilina in California, those thresholds are in development. Qualitatively, we also can use the odor from the treatment. Basically, stronger the unpleasant odor of anaerobic decomposition, the better. But clay soil tends to have less odor. And there are other potential indicators, such as volatiles itself, measuring volatile itself. At this point, it's kind of cost prohibitive, but in the future, it may be able to do it. And iris tubes, this is the PVC pipe coated by iron powder. And you can just bury it in the soil and then take it out after a certain period of time and see how much iron removed. Those areas can tell you how strong the treatment was. But to my knowledge, there's no study correlating with the iris tube measurement and the pathogen suppression level at this point. Another aspect of using carbon source is the effect on fertility, especially nitrogen fertility. For example, we use rice bran as a carbon source. Rice bran contains about 2% of nitrogen. So when we apply 6 to 9 tons per acre, I'm gonna tell you more about it. It provides quite an amount of nitrogen. And then how much actually those organic nitrogen mineralized during crop season is very important. And we growers have to adjust fertility management accordingly. In our case, rice bran decompose at 20 to 30% within 6 months. So basically, by doing ASD, strawberry growers can eliminate the pre-plant fertilizer, especially in organic system. This kind of adjustment is very important. And we came up with the standard ASD approach for strawberries. We call autumn bed ASD. As I said, broadcast rice bran at 6 to 9 tons per acre, which is 13 to 20 metric tons per hectare. Use a compost spreader. And incorporate rice bran and list the beds and cover with plastic mulch as soon as possible. and then drip tapes is underneath. And drip irrigate 1 to 2 acreages, which is 100 to 200 cubic meter. Our goal is to limit up to 2 acreages because that's the amount of a bed fumigate, the water use for bed fumigation. We're trying to limit the water use that level. And leave 3 weeks and monitor soil EH redox potential, and then add water if soil EH increase means system coming back to aerobic too soon, too early. We add more water to keep it anaerobic. And this approach works for Versium dariae, but not for Fusarium oxysporum fragaria and Macrophyna fusariana. So this experiment was done at the Fusarium infested field. Left side picture is untreated control, and for ASD, as like previous approach, it made things worse. But if you do summer, it's much better. And here's the thing: the Fusarium oxysporum fragaria, Fusarium oxysporum in general, is saprophytic, and they actually love rice bran. So we have to feed bacteria to create fermentation before Fusarium feed on it. And to do that, we need higher temperature. That's why summer ASD works, but not the fall ASD. And for summer ASD, we can use cover crop, summer cover crop, when grower can grow, as a partial replacement of rice bran. As I'm going to talk about it later, but the rice bran cost is getting more expensive. So grower can grow own carbon source on the field and use it. And we can get typically 3 to 4 tons per acre of dry biomass. And you can just frail mold it and then incorporate it. And this is how we do it, mowing cover crops. And we don't use cover crop itself as a carbon source, sole carbon source, because that was the original Dutch method, and they don't use it very often anymore because the cover crops CN ratio, carbon content varies depending on the maturity of the cover crop. So instead of using cover crop as a sole carbon source, we supplement with rice bran. For example, we standard method apply 9 tons per acre. And if we have 3 tons of cover crop biomass, dry biomass, we subtract 3 tons from 9 tons and apply 6 tons of rice bran on top of cover crop. So that's what we did here, adding rice bran here. And incorporate it, chisel and rototillin. And we asked Trical, fumigation company, to apply drip tapes and TIF mulch together. This is a fumigation rig, but they modified it and then came up with this kind of system. And drip irrigated. And then we left for, in this case, about a month. It has to be clear top to get the sterilization effect because of the low temperature in our area. But if we use this clear plastic mulch and moist this way, we can reach above 30 degrees Celsius threshold easily, even in our regions. And then control Fusarium. Another example of controlling and suppressing Macrophyna fusariana by ASD is an organic field. Grower standard is like this. And then when we use rice bran 9 tons ASD, it didn't control everything, especially the edge of the bed, but fruit yield, marketable fruit yield was actually doubled. So grower was very happy. But this is mustard seed meal 2 tons, and ASD using mustard seed meal 2 tons plus rice bran 3 tons were not as good as rice bran 9 ton ASD treatment.
You have 5 more minutes.
Thank you very much. Another benefit is the long-term effect of ASD. ASD can enhance soil suppressiveness. This data was taken after 2 years after ASD treatment. We did ASD and grew strawberries whole year and then grew winter cover crop and planted lettuce and harvested. And after harvesting lettuce, we took soil sample, but we still found the ASD plot had significantly lower Vertium diarrhea microscrotia compared to untreated control. And this kind of long-term suppression seems to be related to microbial community shift. Chemical produce, volatiles and organic acids disappeared very quickly. And we found repeatedly that this kind of microbial community shift lasts more than a year. And then detailed mechanism is still unknown, but we think this microbial community is related to this long-term suppression. And ASD itself is not that we don't think it's a silver bullet replacement of fumigant. Especially in organic system, it is always good to combine with other approaches such as crop rotation or use of the resistant variety. In this rotational study, we compared the length of rotation, 2-year rotation or 4-year rotation, which means 1 year strawberry, 1 year vegetable, or that's 2-year rotation. And the 4-year rotation is 1 year strawberry and 3-year vegetables. And we have 2 different rotational crop. Broccoli is known to suppress residue incorporation, known to suppress Vertium diarrhea. Well, this field was infested with Vertium diarrhea to begin with, so we used that. And also lettuce, the major crop in our region, but it's the host of Vertium diarrhea. So we have both. And then for both crop type, we have bare fallow control, the mustard cake, and ASD plus compost. And then we did the 8 years of experiment. And then 8th year, we planted strawberries to all plots and compared the cumulative effect. And for rotational length, 4-year rotation has 20% more yield than 2-year rotation in average. But that's not the case for broccoli. When you have a broccoli rotation, pretty much similar yield, regardless of rotation length. Also, even ASD didn't make much difference, but that's not the case for lettuce rotation. Probably lettuce is the host of vert, so ASD might have helped reduce that negative effect by planting lettuce and increase yield so much, and long-term rotation was better. And this is the average of 10 or 11 replicated field trials across California between 2010 and 2015, different soil type, different pathogens. And then, this is the ASD average relative yield to fumigant is 99%. So pretty close, very close, pretty much the same. And if you add one more trial with highly infested with Fusarium, the average went down to 95, but that's pretty still similar with fumigation control. With that kind of data, growers started to adopt ASD in California strawberry production. And the most recent number I have is about 1,700 acres, about 700 hectares in California, mostly organic strawberries, which represent 30 to 40% of California organic strawberry acreage and 4 to 5% of California total strawberry acreage. And then now as the research is spreading across the United States, many other states, especially Florida and the Southeast states, started to work on this approach. There are some key practices for successful ASD. First one is uniformly mix carbon source with soil. It's very important. And apply mulch as soon as possible after carbon source incorporation to avoid anaerobic decomposition of carbon source and preserve soil moisture and reduce the amount of drip irrigation. And use with appropriate crop rotation. We are working on this kind of study right now as a separate project. And use of resistant variety when available. Limitations, cost of carbon source. In our case, rice bran is getting higher. It costs like almost $3,000 per acre. So at this point, it's been use of ASD is limited to high value crops and nurseries. And we are always looking for alternative carbon sources, and use of cover crop as a partial carbon source can reduce the cost. And use of plastic top, this is essential, but that study trying like a spray top or something, but doesn't seem to work well. So it's still necessary part of the practice. water use. Depends on the soil moisture when at the bed listing and soil type, it can range, but trying to limit that. And again, SD is not super bullet and should be used in combination with other tactics such as crop rotation, resistant cultivar, and grafting when feasible. What's next? Test if interaction of ASD with genotypes exist. We are doing that right now for strawberries. And developing a simple method to measure labile carbon in potential carbon sources. Quality of carbon is important, not just total carbon CN ratio. And evaluate greenhouse gas emission and leaching potential in ASD. Florida studies shows that that's not much, not much more compared to conventional system, but studies in progress in California. And use models to analyze field and greenhouse trials to identify optimal conditions for control for specific pathogens, and then combine with microbial amendments. That's in the future. And use as a part of integrated soil-borne pest management. As the effect of ASD varies depending on pathogens, identification of the pathogen is very important. Thanks to molecular approach development, all three major lethal pathogens can be detected using molecular approach very, very quickly and accurately. So based on that diagnostic, we can choose and then construct the strategy how to control a pathogen. And that's development is very important. With that, I'd be happy to answer any questions. This is a photo of strawberries in here and then Canadian maple in my garden. Thank you very much.
Thank you very much for your presentation. We have 2 more minutes for a few questions. Do you have some, Sean?
Yep, there's a couple in the chat. Have you tried incorporating brassica along with flooding and solarization to reduce inoculum level of subject pathogens? And I think I have a similar, we have tried that in ginseng, I guess. Are those things compatible, trying to control pathogens at the same time as trying to do ASD? Yes, we've been trying mustard seed meal as a carbon source many years, but we need a lot more amount. Usually 2 to 3 tons is a limit because of the cost, way more expensive than rice bran. And then there's a successful example in apples, which they can apply only 1 time in 20 years or something. So they can apply high amount, 3 to even 5 tons or something. But for strawberries and annual crop, it's cost prohibitive, and we didn't find very effective result by using mustard seed meal alone. Okay, when counting Verticillium dahlia's microsclerotia, how much confidence do you have that it is all Verticillium dahliae as opposed to other Verticillium? Well, I didn't do the experiment, but it was a dry seeding method using selective medium by Steve Koike. So he's the expert of Verticillium dahliae, so I'm pretty sure that's the Verticillium dahliae. When you say labile carbon source, how are you determining this if not carbon to nitrogen ratio? What is the carbon nitrogen ratio of rice bran? Rice bran's CN ratio is about 20, 2% nitrogen and 40% carbon. But a labile carbon, those CN ratio is a ratio of total carbon and total nitrogen. And some carbon, like lignin, bark or wood chips, and so speak, it has a lot of carbon, but it's kind of inert form of carbon. The microbes and especially bacteria cannot use as a resource quickly enough to create anaerobic condition. So the type of carbon has to be a labile, biologically available form. So there are a couple methods being proposed. One is to measure a total sugar content of carbon sources. That could be one way to determine if the carbon source is useful as a carbon source for ASD. And could you use digestate from methane generation as a carbon source?
There are some studies I use that, and personally, I haven't tried that yet. But if the material has been digested already, I think again, labile carbon might be very low. So, like we try using compost as a carbon source, it doesn't work because it already decomposed, right? So probably the same thing's going to happen.
Okay, thank you very much for your presentation. I think everyone found it very useful and interesting. Our next speaker is Dr. Sabine Vanessa. She's a professor in plant pathology at the Crop Development Center of the University of Saskatchewan. She will be presenting on the management of root rot complex in pulse crops. Thank you, Sabine, for being here with us, and please share your screen, and we're eager to hear you.
Thank you for the introduction, and good afternoon, everybody. So I guess, oops, switching gear a little bit here to the beginning of my presentation on managing the root rot complex in pulse crops. So ASD sounds very interesting, and it's unfortunate that on large scale crops, it is probably not an option just because of costs. So in pulses, if you have driven across the prairies in the last few years, you've probably seen sort of fields like the picture I'm showing here. And managing this root rot complex is obviously very complex. So why are we seeing more of these root rot pathogens? So probably the primary or one of the really important reasons is that we have been seeing more and more tight rotations. Farms have become larger, and so producers have tried to simplify their crop rotations, often just having a canola, a wheat, a pulse, and maybe another cereal. And depending on the market, they may tighten that up even further to really benefit from crops that are doing well in the market. Connected to that, there is probably a certain level of neglect of soil health. Less diversity in terms of crops often is reflected in soil health and diversity of the microbiome in the soil. At the same time, there is a lack of efficacious chemical or biological treatments, and the varieties that we are growing are primarily susceptible to the primary soil-borne pathogens that are affecting the pulse crops. So what are we dealing with in terms of root rot pathogens? Now in the pea and lentil surveys have shown that it's primarily Aphanomyces eutychus and various Fusarium species. And these surveys that were conducted between 2014 and 2017 under the leadership of Sharma Chatterton across the 3 prairie provinces show that indeed very often it is a complex. So for example, Aphanomyces eutychus almost always shows up in combination with Fusarium. However, Fusarium, there are fields where Fusarium is the only genus that does show up, so no Aphanomyces yet. If you have a closer look at the Fusarium species, in the pea and lentil, the predominant species is Fusarium arenaceum, Fusarium solani, and Fusarium redolens. And then at least in Saskatchewan and also in Manitoba, there is also some Fusarium graminearum. So if you're familiar with cereal diseases, these species will be quite familiar to you as well. In particular, Fusarium graminearum is very well known as the major problem with the Fusarium head blight. What does it look like? Well, if you dig up plants, what you will see is that the root system is stunted. it's usually discolored, and depending on how severe the root rot is, the roots may have turned completely black. You usually do not find any nodules anymore. So not only are these plants hampered by water and nutrient uptake, but they are usually also nitrogen deficient. And that's why you then often see this yellowing as well. So sort of an indication that they don't have functioning nodules that fix air nitrogen. In the rare case that you are seeing or digging in fields that are only infected by Aphanomyces eutychus, you may see the classical symptoms for that pathogen, which is this caramel-like honey brown color, which I'm showing here in this picture. But as I showed in the previous slide, there are only relatively few fields that are only infected with Aphanomyces. Very often these plants are co-infected by Fusarium, and that sort of muddies the water a little bit. What you see more commonly and can be a telltale for Aphanomyces root rot is the deterioration of the cortex. And you can see that in this picture here, where you basically can strip off the cortex and just have the vascular bundle left from that root. If you're very lucky and cut open a plant with the root that has been infected by root rot pathogens, sometimes you can see a red streaking in the vascular bundle, and that is indicative of Fusarium species. But it is not always there, so you have to be a little bit lucky. In chickpea, we've been seeing more root rots as well, and sort of surveys only started, detailed surveys only started last year, and it seems that Fusarium is the predominant species in this crop, at least based on preliminary data. So in this table on the right, I'm sort of showing you what we found in 42 fields in the rural municipalities where chickpeas are grown. This is incidence data, and sort of the most common pathogen was Fusarium solani, followed by Redolens, Arenaceium, and Sporotrichoides. So we are still dealing with or analyzing this data further. We also have more data from this year, so we'll see whether we can confirm that Fusarium is the predominant genus here, and this will be followed up then by pathogenicity testing to determine which among these pathogens are maybe the more virulent species that we should focus on. And then at least here at the CDC, the idea is then to move towards resistance screening. So generally, what management options do you have in the pulse crops? Crop rotation is an important management strategy, at least for Aphanomyces, where right now we really don't have any other tool. But when we talk about rotation for this pathogen, we're talking about 5 to 9 and maybe even longer years without a pea or lentil crop, because the oospores are so resilient and survive in the soil. I put a question mark there because there is really no hard data for our growing conditions to say how long you have to wait. Now, 5 to 9 years without a pea or lentil is not something that growers want to hear, but some growers certainly are reconsidering whether to include pea or lentil in their rotation, simply because their fields are so heavily infested. With Fusarium, crop rotation obviously is less of an efficacious option, simply because many of these species have a much wider host range, and so the rotation will have less of an effect there. One interesting option that has garnered more interest in recent years is cover and intercropping, specifically for Aphanomyces, and the picture I'm showing here is from one such trial conducted by Christopher Morrison, who is a PhD candidate at Canada in Lethbridge and here at the University of Saskatchewan, co-supervised by Dr. Chatterton and me, who is looking at the effect of brassica, rye, and oats, all of which are known to have antimicrobial properties on ooze spores and disease development. I don't have any data yet. Christopher literally sits on a big heap of soil and is still sort of fine-tuning his PCR method to quantify Aphanomyces in the soil, which is very, very tricky. And then there is a lot of research going on biological control here at the university and elsewhere in the province and even sort of across Canada. And I quite regularly get requests for isolates. The latest was from Vancouver, where I always thought they don't grow any pulse crops. Last but not least, and that's sort of where a lot of my time is spent on, are the development of resistant varieties. And I want to focus a little bit on that to tell you what we have been doing in that regard. So in the P, we've been very fortunate in that colleagues in France and at the USDA in Washington State basically did all the legwork for us and did an enormous amount of screening to identify germplasms with some resistance and also to identify or determine the genetic control of resistance. And so they have identified loads of QTL. The last time I counted, I think there were 52. So it's a highly quantitative trait, which makes breeding a little bit more complicated and difficult. But I just show you a subset here, and I highlighted two which have a fairly large effect, so AEPS 4.5 and AEPS 7.6a. And we acquired the sources of resistance carrying those two QTLs and other minor QTLs, and then started a backcrossing program with 7 CDC-P varieties. So these are the two sources here. PI lines and then 90-2079. They were backcrossed to the CDC parents and then went through 2 rounds of backcrossing. And again, because of the work that had already been done in France and at the USDA, we were able to develop molecular markers for the QTLs, so could incorporate marker-assisted selection, which is faster and easier, in particular in the early generations where you have large number of individual plants that you really want to screen. And so we could delay the first phenotyping to the BC2F3, at which point then we selected the individual plants with the best phenotype and genotype and intercrossed those, and then advanced them to the F2. And again, at each stage, did marker assisted selection to make sure we didn't lose any, or to lose as few of the minor QTLs as possible. So this is, and then we at the F2 we did another round of phenotyping, and sort of this is what we have right now. So on the very right hand side you see CDC Meadow, which is always included in all of our tests. It's our susceptible check. The green lines are the CDC recurrent parents. The 2 yellow bars represent the 2 sources of resistance, and then the blue lines are the ones where we combined, where we pyramided the resistance from PI 660736 and 90-2079. And you can see that we have some lines there that perform really well compared to the CDC parents. The caveat here is this is all based on indoor testing. We have developed an Aphanomyces root rot nursery here at the university, but just as many farmers experience, unfortunately, we also have Fusarium there. So that obviously limits the ability to test these lines in the field right now. And what we have to do is sort of incorporate Fusarium avianatium and Fusarium solani resistance there as well. And I think then only will the disease nursery be very, very effective. So we have started with Fusarium avianatium, whereas And at Canada and NRC, they will hopefully focus on the Fusarium solani. So that looks very good. We also spent time to look at the virulence of Aphanomyces eutychus. Again, the French sort of did the legwork here and characterized a large population of French isolates and a few North American isolates, and came up with sort of several pathotypes. Initially, the assumption was that in North America we had pathotype 3, which is considered less virulent. But our research showed very clearly that the population here is dominated by the more virulent pathotype 1 as well. So it's not great news, but there we go. Now, what is a little bit scary maybe is that when we compared the isolates we tested with the standard for pathotype 1, which I indicated here with a red bar, RB84, you notice that we have already in the population isolates that are quite a bit more virulent than RB84. So that all, of course, is a little bit worrisome because nobody really knows how durable the resistance is we have right now. In lentil, we are really at the beginning here. We screened some cultivated lentil germplasm, but then very quickly moved to screening wild species. We've done a lot of work with wild species in the past on other diseases. And our results indicated, yeah, we have some fairly good resistance here in the wild species as well, in particular in Lens orientalis and in Lens lamotii. We were lucky in that among this germplasm, we had 2 accessions for which we already had a mapping population. So rather than developing a new mapping population, we could jump in there right away. We see phenotype that population.
5 more minutes.
Yeah, thank you. We phenotype that population, and it's now being genotyped in Australia using a chip. So in German, you may know that my background is German. In German, we have I guess, a nice term for the root rod complex, specifically in the pulse crops. It's called Leguminosenmüdigkeit, or literally translated pulse crop fatigue, which is something you may experience right now after lunch. So maybe a coffee will wake you up, but obviously solutions for the root rod complex in the pulses is much, much more complex. And even with improved resistance, it will not be the silver bullet. We will need sort of many other tools to manage that disease complex and secure the sustainability of pulses in prairie provinces. And with that, I'd like to thank you for your attention and acknowledge the funding bodies and my research group. Thank you.
Thank you, Sabine, for your presentation. It was very interesting. Do we have any questions for Sabine, Sean?
Yes. So first question, does anyone here, or do you know of anyone, do research on soil-borne fusarium species in peas in Ontario, eastern provinces, becoming an increasing issue in Ontario, but processing peas are a minor crop here?
I am not aware. I know that there was a little bit of research in Quebec, but I'm not aware of anybody looking at this in pea in Ontario.
Okay.
I'm in New Brunswick Project and Research Center. I had one project starting 2018, and then did some field survey on field pea in New Brunswick, and also some field in PEI. And then the main soil-borne disease is Fusarium. That's multiple species there. Yeah, that's the information I could give now.
That's it for questions in the chat. Are there any questions verbally? Oh, there's another one. You mentioned Aphanomycetes mostly showed up with Fusarium species. Any specific Fusarium species that shows up with Aphanomycetes that you know of?
Well, as I showed in the slide, it's Fusarium avianacum and Fusarium solani and maybe a little bit of Redolence as well. So these are probably the most common one, which are also, at least avianacum and solani are also quite virulent as pathogens on pea and lentil.
Okay.
So we don't have any more questions, or in the public, anyone has any further questions, please raise your hand. Okay, thank you, Sabine, for your presentation. It was very interesting. And now we go with our next speaker, who is Dr. Samir Punja. He is a professor in plant pathology and biotechnology from the Department of Biological Science in Simon Fraser University. He will be presenting today in a trending crop, which is cannabis, and he's going to present us about root-infecting pathogens of cannabis sativa and disease management approaches. Thank you, Samir, for being with us today.
Thank you, Andrea, for organizing this workshop, and to the rest of you that helped organize this, thank you very much. It's morning here and it's afternoon most of where you are. It's a great time to start thinking about cannabis, and what I'd like to do today is talk to you about root-infecting pathogens of cannabis sativa, and talk a little bit about the disease management approaches that we're currently considering. And my email is over there if anybody has any follow-up questions. For those of you that are not familiar with cannabis or marijuana, I'd like to give you a quick background. This crop was approved by Health Canada back in 2018, and we currently have about 391 licensed producers who propagate, grow, and harvest the crop. Most of the production is indoors, but there is increasing interest in growing cannabis outdoors, and we currently have about 38 licensed producers that grow outdoors. For the purposes of my presentation today, I'm just going to focus on greenhouse-grown cannabis, which is the predominant acreage at the present time. Those of you not familiar with how cannabis is grown indoors, I'll just quickly go through this slide here. The plants are started from what we call stock plants or mother plants, which are plants that may range from 1 to 4 years of age. And the crop is primarily propagated vegetatively, and so cuttings are made from these stock plants, and these cuttings are then rooted and transplanted into either cocoa or rockwool blocks and allowed to grow for a period of about 2 weeks, after which time they're exposed to reduced lighting conditions, usually 12 hours of light, 12 hours of dark. And what that does is it stimulates the flowering on the plant, which is obviously the part of the plant that's of interest. And then these flowers continue growing over the next 6 to 8 weeks, at which point they're harvested and dried over a period of about 6 days to give you this final cannabis or cannabis bud product. So the entire life cycle is anywhere from 3 to 3.5 months indoors in a greenhouse. Outdoors, there'd be only one cropping cycle starting probably May all the way through September. So when you think about diseases, the locations where we do find many of these pathogens are on the stock plants, on the mother plants that I showed you that are the source of cuttings. We find pathogens on the vegetative cuttings as well, causing things like damping off. We also see pathogens on the vegetative plant growth, in other words, the stage between the cuttings and the flowering. And we also see pathogens in the flowering stage. So essentially from start to finish, we're seeing a range of these different root infecting pathogens. The planting medium that growers used is either cocoa fiber or rockwool, or a combination of peat, cocoa fiber, or in some cases soil. What I'll discuss today is mostly propagation in cocoa fiber and rockwool, which is probably about 90% of the production. And the major pathogens I'll talk about today are Fusarium and Pythium, which you've heard a lot of already this morning. So here's a very healthy stock plant or a mother plant, as you can see. And it's from these plants that vegetative cuttings are obtained. These cuttings are then transplanted, as you'll see in a second. But these stock plants, these mother plants, are susceptible to infection. And what you see here is Fusarium root and crown rot. And you can see the devastating impact of this crown lesion here on the plant itself, which is completely yellowed and wilted. And in this particular image is dead. So clearly, these plants are not going to be ideal for provision of cuttings for propagation. Here's what the Fusarium, in this case, Oxysporum does. You can see the discoloration in the xylem and the pith. And these plants are obviously not taking up enough water and nutrients. And so they start to yellow and die, as you saw in the previous slide. And from these tissues, we get pure cultures of Fusarium oxysporum. Very, very high incidence of mycelial growth and spores in these diseased plants. Now, in the studies that we've done, we've actually shown that the Fusarium infection can spread up in the plant for distances of about 200 centimeters. So you're looking at a plant that's about 2 meters high, and yet you've got evidence of Fusarium infection internally. And that has a big impact, as you'll see, on cuttings, because clearly cuttings that are taken from these diseased plants are going to transmit the pathogen. And in fact, when you do isolate from tissues 200 centimeters from the base or from the crown of the diseased plant, you do find Fusarium presence as high up on the plant as indicated. And so what that means is that when we start propagating from cuttings, and you see here a nice propagation room that has rock wool, and these cuttings are taken from the mother plants and inserted into the rock wool cubes. If everything goes well, of course, these plants will root and continue to grow. But in the presence of Fusarium, as you see here, we get what's called a damping off, where you can see infection right at the base. And there's evidence of sporulation right here on the stem. And again, when we isolate, we get pure cultures of Fusarium growing on PDA. And we have some pretty good evidence now that the initial source of infection or inoculum was actually the diseased mother plant, which actually may be asymptomatic. And so when cuttings are taken from those plants, that immediately gives rise to this transmission of disease and damping off. The disease will progress. If it passes through the propagation stage, you will see symptoms at the vegetative stage. So here again, these are plants that potentially made it through in the propagation, but now are showing symptoms in the vegetative phase. And you can see the classical symptoms of yellowing and wilting, and in this case, complete death. The disease will progress. It'll go into the flowering plants, as I mentioned earlier. And so on the flowering plants, you see the typical Fusarium yellows symptoms, if you wish, where you've got the yellowing of the foliage and the plants are stunted. And if this disease continues, then it'll transmit all the way through the plant. And here you see isolations that are made higher up on the plant, towards the top of the plant. And you can see evidence of recovery of Fusarium from all of these tissues. So clearly, when the plants are this diseased, they are going to collapse. And on a very hot, sunny day, you will see plants that are infected by Fusarium showing this yellowing and in some cases, complete death. You can see healthy plants in the back there. So this is clearly a hot spot or a focus of infection due to Fusarium. So in order to determine to what extent Fusarium and Pythium are present in these indoor production facilities, we conducted a sampling over 3 years from 2018 to 2020. And we went through the standard surface sterilization and isolation and identified pathogens that were recovered by PCR of the ITS1, ITS2 region. And we also conducted pathogenicity tests, as I'll describe. We surveyed 7 production facilities and sampled more than 150 plants throughout this particular study. So here's just a quick overview. We take the diseased plants, we sterilize them, place pieces onto agar medium, PDA with antibiotics, get the fungi to grow out, subculture them, and then identify them after DNA is extracted using the PCR. And then we conduct the pathogenicity tests. So about 2 to 3 month process going from isolation all the way through testing for pathogenicity. What did we find? In the case of Fusarium, we found Fusarium oxysporum to be the most common pathogen, 70% recovery. Fusarium proliferatum, another species, about 15%. Fusarium solanae, about 5%. And then a very low incidence of this other Fusarium like Inoculum, which is sort of a cylinder-carpent type organism that we found once in 2019. So the predominant species are oxysporum and proliferatum. And when you see these cultures, you can see the range of variation that we find with the Fusarium oxysporums and the Fusarium proliferatums. Most of these infections, as you can see, are here at the crown, or in some cases on the root themselves. And you do get quite a bit of sporulation here at the crowns of the diseased plants. And we do see these spore clusters, as you'll see in most Fusarium oxysporum type fungi. And these obviously will spread. They'll spread within the growing environment. And we have evidence to show that these things not just spread in water, but also in the air. And so what happens is you do get lateral spread, obviously from root to root, because these are in hydroponic systems. So you've got inoculum flowing laterally. But interestingly, we do find inoculum in the air. There's a lot of air movement in these greenhouses, and the spores can be spread upward onto the plant. And I'll show you some evidence that indicates that although we're dealing with the root rot fungus, you can actually get infection of the flowers, causing a bud rot, which is quite unusual, although it's becoming more common now. So here's a Petri dish assay. We lay out these Petri dishes in the growing environment for an hour, bring them back, and after 6 or 7 days, we evaluate these plates to see what types of fungi are growing. And as you can see here, colonies of Fusarium oxysporum are present in the air that are now showing up on these Petri dishes. And more so is this devastating symptom where the Fusarium, in this case, it happens to be Fusarium graminearum, is actually on the flowers, causing a bud rot, very similar to what you see in wheat head blight, except this is cannabis bud rot caused by Fusarium. Not just graminearum, but recently we've also found Fusarium sporotrichoides and Fusarium oxysporum and Fusarium solanae. So there's at least 4 species that cause this devastating bud rot, which is of concern to growers now. We've done some scanning electron microscopy to show that this mycelium, as you see here, completely covers the bud. These are the trichomes of cannabis, and you can see the mycelium just filters all the way through. And despite the fact that things like graminearum and sporotrichoides are found on cereals and other plants, they do very well on cannabis flowers. They seem to flourish in this environment. We did some pathogenicity tests, the classical pathogenicity test using cuttings that were dip inoculated in Fusarium canidia. We also used vegetative plants that are wound inoculated for 5 minutes with a spore suspension. And then we grew these cuttings and plants either in rock wool or in the cocoa fiber substrate, which are the 2 main growing substrates for cannabis. And as you might expect under these conditions, this is the inoculation of the cuttings. And you see the mycelium just traveling right up the stem here, causing the damping off that I showed you earlier, as well as the yellowing and necrosis. So this is a devastating disease on these cuttings. And in addition, when you have these plants in soil that have been inoculated, you see the typical symptoms of yellowing and stunting compared to a control on your left. So these Fusarium species are indeed quite aggressive on the roots, on the cuttings, and also on the flowers. So just to summarize what we found with the Fusarium, Fusarium species that are causing these root and crown rot diseases on cannabis are not wilt pathogens. And at the present time, there's no evidence that they are forma specialis of Fusarium. They seem to be more general broad host range fungi. Major source of inoculum were diseased mother plants or stock plants, which in many cases are asymptomatic, but when you take cuttings from them, the disease is spread. And then finally, the inoculum can be found in the air as well as in water, and it spreads throughout the production facilities. And in many cases, it may also infect the flowers. Now, one of the production systems that's used here is quite interesting. in that when the vegetative plants are grown on these benches, twice a day they're flooded. As you can see here, the taps are turned on for 15 minutes or so until you've got about 2 inches of standing water on these benches, and then the drains are open and the water is released. This is a perfect environment for both Fusarium and Pythium to grow and spread. Unfortunately, that's the only way they can currently irrigate these plants is by flooding them. But I think, as we all know, this creates ideal environment for these pathogens to survive. And in fact, when you lift up some of these diseased plants, you will see that the roots on the bottom here have completely turned brown, which is a typical symptom for both Fusarium and Pythium, less so here on the left, where the plants seem to have escaped infection. And of course, in a really severe case, just about every plant on that bench has been infected by either Fusarium or Pythium, as you'll see in a minute. And you can see the symptoms of yellowing and stunting. These plants aren't going to make it. They have to be destroyed and start over. Okay, moving on. We also find Pythium when we isolate for Fusarium. So you see here a colony of Fusarium, but you notice also a very prolific growth of Pythium as well. So the next part of this presentation is going to be characterizing some of the Pythium species. And they cause similar root rot and wilt. The symptoms are a bit different. You get more of a complete destruction of the root system here with Pythium and a more intense yellowing early on than you do with the Fusarium.
You have 5 more minutes.
Thank you. As you see here, the symptoms are quite dramatic. On the right is a plant that's wilting due to Pythium infection. And these plants will continue to collapse and rot as the pathogen progresses. So here you see complete destruction of the root system and complete browning here, more so than you would expect with Fusarium. And here again, on a hot sunny day, the plants literally collapse. So you've got complete rotting of the root system, much more severe disease than you expect to find with the Fusarium. And of course, isolations from these tissues will give you pure cultures of Pythium as well. We've seen a lot of the mother plants, the stock plants that have received water-saturated conditions where the roots are brown. We get lots of Pythium coming out of these diseased tissues. So what are we finding in terms of species? Pythium myriotylum, 48%, Pythium disoticum, 30%, Catanulatum, 10%, Ultimum and Ephinoddermatum, 5 and 7%. So we've got 5 different species of Pythium that potentially can cause this problem on cannabis, with myriotylum and disoticum being the most important. And again, we've done these pathogenicity studies that show that when you inoculate with a mycelial suspension, you do get stunting and dwarfing of these plants compared to the controls that don't have any inoculum. So these are indeed aggressive pathogens. So inoculum sources, as I mentioned, disease propagation of propagated plants. There's inoculum in the environment, hydroponic solution on the benches. In the growing substrate, they could potentially be spores of Fusarium. And there's probably evidence to suggest that inoculum from neighboring plants may also be able to infect cannabis. So there's multiple sources of these pathogens. What can we do about Pythium and Fusarium? Diseased stock plants should be destroyed. Oversaturation of the growing medium should be avoided. Vegetative plants with symptoms obviously should be destroyed. There is no evidence that there is any disease resistance in cannabis strains. And we have some evidence to show that some of these biological control agents, such as pre-stop and root shield, can also provide protection against these two pathogens. And so these are the 2 fungi that we're investigating a little bit more now. This is pre-stop on your left, and this is trichoderma or root shield on your right. And these are both registered for management of these root diseases. And if time allows, I can go through and show you some data, but there is certainly good evidence that when these 2 biocontrol agents are applied early on, they do provide some pretty significant protection against infection by these 2 fungi. Now, this is just a list of the overall types of biocontrol and pest management products that are available for cannabis. The list is not extensive. There's about 10 or 11 of these. Many of these are biological control products. There's vaporized sulfur, but notice there's no fungicides. There are no fungicides registered on cannabis. And so growers are left to using reduced risk products or some of these biological control agents such as trichoderma and Gleocladium or bacillus. So you have to use a series of management strategies such as sanitation to manage these various pathogens. So I'll just quickly go through a couple of slides and show you that we've done some biocontrol studies where when you add the biocontrol agent to these rock wool cubes, and then you insert the cuttings and then inoculate the cuttings with fusarium, you do see a fairly significant protection. So here's an illustration. On the top is the control, and on the bottom is a rock wool cube that was flooded with pre-stop, and then the cuttings were inserted into the rock wool cube, and then fusarium was added, suggesting that the presence of the pre-stop was actually inhibiting the colonization of the plant by fusarium. And here's some data to support that. When you look at the incidence of disease, you notice with pre-stop and root shield, the amount of disease that you get is significantly lower than it is in the control. And so these two biocontrol products are providing a fairly significant protection against fusarium. This is a bacillus product, Rhapsody. Not as good, but did see some reduction that was not significant. So adding these biocontrol agents early on seems to provide significant protection of the cuttings. We also repeated the experiment with Pythium. With Pythium, it's a little bit more complicated because you've got to get roots to form. So you get the roots to form. As you see here, you add the biocontrol agent, you wait for a week, and then you wound the roots to provide wounding sites for the Pythium, and then we inoculate. So a little bit more complex than just inserting the cuttings into rock wool. We had to root them first, apply the biocontrol, and then induce some type of wounding. And when we did that, what we found as well is that both root shield and pre-stop significantly reduced disease compared to the control. So again, a preventative application of either biocontrol agent followed by inoculation was significant in protecting these rooted plants against Pythium. And so finally, my last two slides, we have some evidence now that these biocontrol agents are actually endophytic colonizers. In other words, they can get into these cuttings and establish themselves internally, which sort of makes sense if they're going to provide some protection against these pathogens, both Fusarium and Pythium. So summary, these pathogens are presenting challenges to growers. These biocontrol products that we looked at appear to be quite significant in terms of the protection they offer. Sanitation is going to be quite important. We talked about inoculum movement and the spread of pathogens in water. I haven't said much about anything other than Fusarium and Pythium because there are no other reports of these other fungi, although diagnostic labs tell me that Verticillium, Phytophthora, Razoctonia, and Phalaenopsis are present on cannabis, but there's been no research done on it. So we're awaiting incidents or outbreaks of these pathogens before we can actually conduct more experiments. And with that, thank you to the funding agencies, to NSERC, to the BC Ministry of Agriculture, Canadian Agricultural Partnership, and many, many students and technicians that have helped me conduct this work. And thank you for the extra few minutes to finish my talk.
Thank you, Samir, for your presentation. I think it was very interesting. So we have a couple minutes for one or two more questions. So.
Yeah, so there's a few in the chat. Do you have any record of resistance in cannabis against these diseases?
There isn't any that I know of, although there's some interest in looking at these land races. These are the sort of the progenitors of cannabis that were growing in the wild many, many years ago. But to my knowledge, there's no evidence at all for resistance.
Okay, is in vitro propagation a viable and cost-effective alternative to avoid Fusarium propagation in greenhouse crops?
Yeah, that's a good question. So the use of tissue culture is being looked at extensively, but it's more to avoid viruses and viroids than it is for Fusarium and other diseases. I think you can manage those through proper sanitation and other methods rather than in vitro culture. I think in vitro culture will be very effective against viruses.
Okay, Fusarium oxysporum is a common soil pathogen. In your opinion, what are the reasons that this crown rot has not been observed, at least to any extent, in outdoor cannabis production in Ontario, for example? What is the optimal conditions for the infection?
Okay, so that's a good question. And just like cannabis starting indoors, the more you start growing outdoors, the more diseases you see. So there actually is a report, several reports of Fusarium oxysporum on outdoor cannabis, as there is for many Pythium species grown outdoors. So it's just a matter of time, I think, before these crops start to show disease symptoms. And it's not surprising that we find both Fusarium and Pythium outdoors in soil. They're prevalent regardless.
Was wondering if combining two species of trichoderma, such as Asprello and root shield, at the same time can provide any better protection? Yeah,
that's a good question. And growers, as most of you probably know, are quite creative in some of the things that they do. And they do try combinations of biocontrol agents. To my knowledge, they haven't shown any extra additional efficacy. Many of these work similar fashions. They're either endophytic or they produce enzymes or so on and so forth. My worry sometimes is that they may outcompete each other. So if you've got two trichodermas, they may actually not show significant activity as opposed to one by itself.
Okay, thank you very much. That we have an additional question, so if you can help us answering it on the chat, that'll be very useful. Thank you very much.
Thank you.
So we're going to proceed with our last speaker of this afternoon, who is Dr. Khalil Almurabi. He is the provincial potato pathologist for the New Brunswick Department of Agriculture, Aquaculture, and Fisheries, and he's as well an adjunct professor at Dalhousie University. He will be presenting on potato early dying, research update, and disease management. Thank you very much for being here with us. So please share your screen and we want to hear you.
Okay. Trying to share my screen.
The bottom?
Yeah, I did, but it's just not showing me.
Okay.
Screen.
Okay.
Can you see this?
Yes, we can.
All right. Don't know what happened there, but all right. Oh, okay. A lot has been discussed about verticillium and early dying. Hopefully I can put a few things together and summarize some of that in the work that we've done so far in New Brunswick. So potato early dying is a vascular disease which results in premature vine senescence, and it can limit potato tuber yield by as much as 50%. It's primarily caused by the fungus Verticillium dahlia, but co-infection of potatoes by both Verticillium dahlia and the root lesion nematode Brachylinca species can greatly increase the severity of the disease. In this slide, you see the picture on top right is a potato stem that is infected with verticillium, and it has microsclerotia, which has been mentioned before. On the left, it shows the root lesion nematode attaching itself to the hairy roots of the potato plant. And on the bottom, you see a field that is infected with early dying. How does it survive? How does verticillium survive? It survives in the soil as microsclerotia, which is the resting or overwintering structures. Or in plant tissue, it survives as mycelium or microsclerotia in the potato tubers, leaves and stems. And you see pictures here on the right and the left with microsclerotia on the stem itself, and in the middle, a leaf that is infected, showing symptoms of early dying. Verticillium invades the xylem tissues and disrupts water transport in plants, and then causes foliar discoloration, necrosis, and vascular wilt. And it does persist in the soil for more than a decade as a microsclerotia, which is the overwintering structure. But once it's introduced to a field, the fungus is spread by runoff water, cultivation, and machinery, such as harvesters, which transport soil from one part of the field to another. There are various references on the economic threshold for verticillium, and it varies between 5 to 30 microsclerotia per gram soil, and some put it in the teens. Verticillium has a wide host range. Almost all dicotyledons or broadleaf plants are susceptible to it. That includes potatoes, alfalfa, clover, sunflower, cabbage, and you could read the list there: celery, cucumber, and so on. Monocots, however, which are cereals such as barley and oats and wheats, and also forage grasses, are non-hosts. Asparagus and lettuce are both moderately resistant to verticillium, while none of the potato cultivars registered in Canada is fully resistant or tolerant to potato early dying. What to look for in the field? Symptoms of verticillium include yellowing, wilting, flagging, and discoloration. So yellowing, first symptoms usually appear after flowering on the lower leaves, and the area between the leaf veins and turn yellow, and later on the symptoms move upwards to the younger leaves. Initially, one side of the leaf, as you see in both these pictures, one side of the leaf shows symptoms of yellow and wilt, and later on the whole leaf turns yellow, and the plant also can show browning and necrosis later on. Wilting, not all the stems from the same plant show symptoms. Some do, some don't. And you see the symptoms during the day, but the plants or the disease recover at night. The crop senesce early, about 3 to 4 weeks before reaching maturity. And we see that in heavily infested fields. This year, for example, I've seen that in Russet, Burbank, and also Shepherdie potatoes. Now that will mean that the crop does not reach maturity on time. The size of the tubers will be small and the yield will be impacted. Flagging, as you see the picture here, is another symptom of early dying, where the plants stand higher than the rest of the plants in the field. And discoloration too. So the lower portion of the stem, as you see in the picture on the left hand here, I put some circles on stems that show microsclerotia, black discoloration on the stems. Now, if that stem is still alive and you make a cut in it, what you see is brownish discoloration in the vascular system, as you see on the picture on the right hand. There's also a tuber symptoms, discoloration in the vascular ring area, and that starts at the stem end usually, but it doesn't go beyond halfway through the tuber. The second culprit of the potato early dying complex is the nematode, the root lesion nematode, which can be present in the soil, on the roots, or in the roots and the tubers. Nematodes are microscopic roundworms that attach themselves to the roots and feed there. They then cause damage and stunting, yellowing, wilting, poor stand, also yield reduction. Symptom severity is nematode density dependent, which means the higher the number of nematodes per kilogram of soil, the more severe the symptoms can be. Plant parasitic nematodes, usually including root lesion nematode, do not migrate too far from the root zone. So they go within 1 to 2 meters from the root zone that they infect. And they cause lesions, as you see in the picture on the right top there. Reddish, brownish lesions on the roots are some of the symptoms that you can see. And on the left top, you see a picture of root lesion nematode inside the tissues of the potato root itself. They also have wide host range, but it's a little bit different. Now, grasses and cereals are hosts for root lesion nematode, but not as much for verticillium. Field and vegetable crops can be hosts, and also weeds. But the preferred hosts for root lesion nematode is potato, corn, carrot, sugar beet, veg, soybean, raspberry, and strawberry. Canola, forage pearl millet, Sudan grass, broccoli, and onion are cited to be non-hosts. And the economic yield threshold for root lesion nematode is between 1,000 to 2,000 nematodes per kilogram of soil. 1,000 nematode for the potato variety is superior because it's very susceptible, and 2,000 for the rest of the potato varieties. Now, some of the research that I have done in New Brunswick included surveys. Over the years, we started in 2016, and we surveyed 163 fields, and they were fields of various crops. They weren't only potatoes. It's just so that we know what the status is in rotation crops. We did some 17, 2018, and 2019, and then we refined what we did, where we started taking samples at different times and also from the plants themselves. Now, some of the results that I will share with you, they're just a synopsis of what we did, but thought to focus on a couple of things. Soil sample collection is very important when it comes to potato early dying to get meaningful data and results. The soil samples that we collect them from the field following a zigzag pattern, and we use a soil probe that's 2.5 centimeter in diameter, driven about 25 to 30 centimeter into the ground, or whenever you can't go any further. The number of subsamples we collect depend on the field size. Generally speaking, we go with 1 subsample per 10 feet. We put those in a bucket, we mix them together, and we have a representative sample. Part of that sample goes for testing for nematodes, and the other goes for testing for verticillium. So verticillium extraction is done, and identification is done on air-dried and sieved soil. So we air dry it, and then we sieve it 2 millimeter pore diameter of the sieve. We freeze it until testing is done. And testing is, I sent it to the ACS lab in Fredericton, New Brunswick, where they do molecular testing, and they determine the number of cells per a gram of soil of Verticillium dahlia and Verticillium alboatrum. Lately, I've been also adding culturing, which gives me more maybe comfort myself, on specific media called NP10. And by that methodology, then we get the number of microchlorotia or colony forming units per gram soil, as you see in the pictures on the bottom there. For root lesion nematode, the soil is not dried or sieved or frozen. It's just used as is. I sent it to the University of Guelph. They do nematode identification and count per kilogram of soil. I believe they follow Perman funnel method, which is shown on the left hand of this slide. Some of the data that we've gathered so far. This is from 2016, the 163 fields. You see on the bottom here, various crops, and this is number of root lesion nematodes per kilogram soil. The higher numbers are marked by arrows there. And so corn had a really high number, and so did clover and barley, oats, potatoes, ryegrass, and soybean and wheat. And again, if we go back to the literature, these were cited to be hosts for root lesion nematodes. So we were not really far away from what is going on and or have been going on and cited in the literature. In case of Verticillium dahlia, here I'm showing the number of cells per gram of soil using qTPCR. And again here, the same fields, potato had the highest level of Verticillium dahlia, and so did clover. And these two are cited or in the literature to be favorable hosts for Verticillium dahlia. I thought to throw this slide in because I think it's very important, because soil sampling determine whether you know what's going on in your field or not, and if there are any variations. Selected here 4 or 5 fields in New Brunswick, and we took soil samples from various spots or locations within each field and tested them for both root lesion nematode level and also Verticillium dahlia. And I'm just going to look or focus on the top left one, which says WC Spring 2018, just as an example. So if a grower took a soil sample from the location WC8, they might have found some Verticillium and no nematode. And if they sample WC2 location, they would be happy to say, well, you know, the field is clean. There's no Verticillium, there's no root lesion nematode, which is not true because the distribution of the nematodes and Verticillium isn't consistent within a field usually. WC7, as you see there, has both Verticillium and nematodes in high level. So sampling is important when it comes to determining what's going on with potato early dying in your field. I'm a part of the Canadian Potato Early Dying Network, a network of very large number of scientists from across Canada working on, you know, understanding the potato early dying disease complex from different aspects. One of the aspects that we're studying is the efficacy of various pest control products against nematodes and Verticillium. And this experiment that's shown on this slide, or most of it at least, the first part 1 to 7, is repeated in Ontario, Quebec, Manitoba, Alberta, New Brunswick, I believe PEI too. So we're testing various products to see how efficacious or effective they are against early dying. These include the control, the fungicide Elatus, which is a new fungicide registered for the control of or management of Verticillium wilt by Syngenta, Vellum Prime, a Bayer product registered for the management of nematodes. Serenade Soil is a biological control product. Bayer feels that it may be effective against early dying, and so the network decided to add an experiment on it too.
You have 5 more minutes.
Okay, Senator PSBT is a seed piece treatment, and we had Elatus and Vellum and Serenade mixed together. I was lucky enough to also have a grower that had VAPAM, and he added it to his fields and did a field scale split fields treatment to see the effect of VPAM on early dying, and he had two very different varieties. One is Russet Burbank, and the other one was Snowdon. So these are some of the results that I had this in 2020. I'm just going to go just for the sake of time. Vellum reduced the number of root lesion nematode by 58%. Serenade soil, lucky enough, 79% reduction in root lesion nematode numbers. And Senator was effective and reduced the number by 68%. When it comes to Verticillium dahlia, Elatus reduced the level by 53%, Vellum by 73%, and the combination of both was also effective and reduced the level by 33%. The effect on marketable yield was variable, but most or all the treatments had positive effect on marketable yield. Elatus, 7.6% increase in marketable yield, 8.4% Vellum, Serenade, almost 10%, 25% Senator, and Elatus and Vellum 26%, 27% Elatus, Vellum, and Serenade mix, although they didn't have effect on the nematodes and numbers and the verticillium level, but they did on marketable yield. And here's my field scale results from the growers' fields. Russet Burbank field treatment with VPAM increased marketable yield by 20%. In case of Snowdon, increased it by 10%. The grower seems to be happy. He's repeating this again in this year. We also experimented on chloropicrin, which is another product for fumigation. Here we have 8 fields. We took samples before application and after application. And generally speaking, reduction of the root lesion nematode number was up to 99%. Same trend with Verticillium dahlia. The level was reduced by up to 89 or 90% in one of the fields. Threshold for Verticillium, I did mention what the literature said, but my colleagues Matthew Singh, Tyler McKenzie, and also Daho Chen in Fredericton did an experiment where they inoculated soil with various levels of Microsclerotia, and they planted potatoes and then assessed the above ground biomass. And as you see on the right hand, the above ground biomass was reduced by 30% when 5 microsclerotia per gram soil was added, and 1,000 was very effective in almost killing the plant, as you see in the picture on the left. So conclusions: soil treatments with fumigants and fungicides, nematicides were effective. Soil treatments increased marketable yield, but the levels are very variable. Rotational crops are hosts. 5 microsclerotia per gram soil was enough to reduce the above ground biomass by 30%. And growers should be careful when they sample soil, the fields, and sample them properly to give them the right information. And finally, how to manage potato early dying. As with all diseases, for plant diseases, plant clean disease-free seed, remove crop debris where the verticillium overwinters, long rotations with non-host crops because the microsclerotia of Verticillium dahlia can remain viable in the soil for more than 10 years. Increasing soil organic matter is important. Successive years of non-host green manures decrease disease severity. Plants in the family Brassicaceae, like mustard, Caliente mustard has been used a lot, decrease soil population of verticillium when it's used in a rotation. And it works by either releasing toxic compounds in the soil or by increasing the microbial activity. Host resistance, no resistant potato cultivars are available, but Gold Rush and Ranger russets are tolerant, which means they're susceptible but they don't suffer yield loss due to infection by Verticillium dahlia. Chemical control, mentioned these fungicides and that are effective, and nematicide. Chloropicrin and Vapam worked really well. We've done a lot of work on this to see their effect in next seasons and so on. And all what I can tell you for now, because of the time, that they do really reduce the number of nematodes in the soil drastically. They do also reduce the level of Verticillium in the field in the year you use it, but the population often builds up in subsequent potato crops when they're planted in the rotation. Funding from Canadian Horticultural Council and Agricultural Canada is appreciated, and assistance from agrochemical companies, Dave Bell, also the farmer Sean Paget was amazing in this experiment. Technical assistance of Soyan Shim and the contribution of Dr. Singh, McKenzie, and Dahoshin are acknowledged. And if there's any time for questions, I'll be happy to answer.
Hi, Khalid. Thank you for your presentation. It was a very interesting one. And yes, we have a couple minutes for one or two questions, I guess. So do we have any, Sean?
Yes. So what is the distribution pattern of the root lesion nematode in various regions of New Brunswick?
I'm not sure what you mean by pattern, but the numbers are high and variable. So depending on what's used in the rotation crops determines the level of it. But we've been surveying this for the last 5 years. The numbers still high, haven't really changed, if that would answer your question.
There are a couple of messages to say excellent presentation. So I just want to pass that on.
Okay, thank you.
Question from me. The fumigation, was that done just in the bed or as a banded application?
The fumigation for the Verticillium, sorry, for Vapam.
Well, by either Madam Sodium or Chloropicrin.
Yeah, Vapam was broadcast, which was added on top and then mixed to the soil. Chloropicrin was injected in the soil.
But is that just in the band of the planting row or the whole area?
Chloropicrin was just in the band in the row itself, but the Vapam was applied to the whole field. It wasn't okay. In rows.
Another question in the chat, can nematode population bounce back after treatment?
They do after maybe a couple of seasons or years. Yes, they do. And I do have figures that show all of that for future reference. Okay.
What is the best time to do sampling for nematode population density?
October, November. When the best time for sampling is after the plant dies, because that's when the nematodes start migrating back to the soil. And if you do it before, then they will be inside the roots and you won't really know the accurate level there. So we usually wait until October, like late October, early November. When it's cold, it's okay too, like up to when it starts snowing.
Okay. What is the, okay, so can soil tag on potato carry the nematode inoculum to distant fields?
Can you read that one more time? I don't know if I understand it.
The question is, can soil that's carried on the potato carry the nematode inoculum to distant fields?
Yes, they can. Absolutely.
In your opinion, can you always isolate Verticillium from potato stems?
Not always, but you can. Most of the times we actually were able also to extract it from leaves themselves.
Okay, I think this is our last question here. So thank you very much, Ali, for your presentation. It was very good, very interesting, something different. And now we can proceed for the last part of our session, where we're going to have a broad discussion about soil-borne diseases, which is the topic that brings us out today.
Yeah, so feel free to turn on your cameras, and hopefully most of our speakers are still here if there's further questions. There were a few questions that we didn't get to, so we can get to them if we're running out of discussion points. But I have a question for Joji, which would be, I don't know if you watched Mary Ruth McDonald's presentation on clubroot, but she has seen just the use of the totally impermeable film on organic soils caused a lot of effect that you wouldn't expect just from a solarization effect. And we have seen very similar results in ginseng, although our the heating in the ginseng is a lot higher, so we might have gotten a solarization effect. But how much of an ASD effect can you get just from the amount of organic matter that's already in the soil, especially when in Mary Ruth's case, where it's on organic soil that's 60 to 80% organic matter?
Wow, I have no experience with that, with that kind of soil, so it's hard to say. But again, it depends on quality of carbon that you have in the soil. If there's any labile carbon, it's very likely to have anaerobic condition under the top. But that's very interesting.
I have an additional question for all our speakers. We as soil-borne pathogens researchers, and also like from the point of view of growers, we have to face a major challenge, which is the disease sampling and the pathogen sampling in our fields, so we can get a good idea of what's going on in the field. Most of our diseases have patchy distribution, so we need to know where it is to go for a good decision making. So which ones do you think are the most important challenges that we have to face, and how do you think we can solve it, and how can we make it not only efficient for research but for our farmers as well?
Anybody want to step in there?
I could try it.
Okay.
Your best bet is aerial monitoring. We've been flying cornfields and various other crops using drones, and you can pick up the distribution of pathogens and disease beautifully. It's almost a 97 to 98% correlation. And it's now an inexpensive method, and a lot of companies are offering it. We do all our soil sampling that way too now.
Okay. Do you use like any spectral imagery, like any special wavelengths to collect these images, or just simple visible spectrum?
You're muted.
Always this muting thing. Huh? Many of the cameras have now at least 5 wavelengths that you can use. And in fact, there is a whole bunch of literature available on specific spectra for specific diseases like late blight or even nematode populations, etc. You can check those out. We're doing cameras for greenhouse production for things like mildew, and bacterial diseases and whatnot. And these cameras are just going back and forth in a greenhouse looking at various spectra for specific disease.
Suppose one of the challenges there is the background research to begin with on especially some of our more obscure crops that have lower acreage to correlate those with the actual pathogens.
It's coming along in leaps and bounds. I mean, there's a huge number of people getting into this.
Yeah, but George, I think you're a little bit optimistic in terms of those assessments. My experience, from reading those same papers and listening to presentations at APS conferences where there's a lot of that work is going forward. They often can identify areas that are stressed, but they can't separate the effect of a big rock under the soil from the effect of late blight on potato, much less differentiate between early blight and late blight on the same crop. I really think you're overstating the ability, where we're at with the actual technology.
I tend to agree with Bruce. I do have a PhD student who's working on potato diseases, and he's using cameras. And he's sending me some of those pictures to look at, and it's really hard thing to determine just from images where the problems are and what is causing them, like Bruce said. It's a good tool, but I don't think it's that tool.
Well, it's kind of ironic that, Bruce, you showed a picture of an area of dead plants that were infected with the clubroot. You don't think a camera can pick that up?
Yes, it absolutely can. Then we showed it with the picture, but you can't tell the difference between that and problem with a herbicide application or some other stress factor.
You may have to get down to the field to find out whether it's a rock or some other issue. But if you do get down to the field, you'll be able to get to those sites that are stressed for what reason, we don't know, but you'll at least be able to map that field for the stress factors. And it could be water.
Yeah, I agree, but we're not at a place where we can differentiate diseases or differentiate stress at this time. There's lots of technology using AI with hyperspectral analysis that has real potential, and in fact, Mary Ruth and I are doing some of that work even now. We're going to working with specialists in that area, but it's not there yet. And the guys that are flying crops and saying that they can differentiate disease from other factors are just telling you things that aren't really true. They can differentiate healthy crops from stressed, but they can't differentiate diseases from other factors.
I agree. I say from a point of view of diagnostics, it's a good combination. I think everything like that has its place, but I still believe that I agree that you can't tell from a photo the difference between a disease or a rock or excessive moisture in a field or a nutritional problem. It all has to do with going to the field. It may be an indicator, hence they go hand in hand.
Yeah, I guess on the actual sampling side of things, if we do have to measure the inoculum in the soil, the challenge growers face is, I deal with ginseng where they're always moving to new land, and they would like to be able to make decisions on whether they can grow ginseng on that land based on inoculum levels. But these things are so patchy, and how many cores do you need to represent the amount of inoculum in the soil, and it becomes, for some pathogens, so many different cores that it would just not be economical to do that. Do we think there's more quantitative techniques coming out, or is sampling always going to be a problem?
I agree, but generally speaking, the larger the number of soil samples taken, and I showed this zigzag pattern. I mean, the literature can show you, in small fields, which pattern you should follow. Is it W or is it zigzag, and so on. But the larger the number, and using the correct methodology of soil sampling really pays off, because I do have growers that take soil samples incorrectly, and then they don't get the results that they assume correct. So it's really important.
So there were a few questions that we didn't get to. First was for Mary Ruth. What was the strain of Fusarium that was in your spinach trials? You're on mute.
Of course. It was Fusarium oxysporum forma specialis spinaceae. So it's specific for spinach.
The next question was for Zamir. Pythium, the Pythium in your trials, was the source of the Pythium the mother plants or external?
Yeah, that's a good question. We don't think the Pythium is actually originating from the mother plants. We think it's either external in terms of the water source, depending on whether it's a well or whether it's city water. But in this particular facility, the water was being recycled. And as most of you know, in a greenhouse production environment, it's very hard to take water and completely sterilize it, whether you use UV or ozone or whatever method you use. And so we think the Pythium has been building up in the pipes and in the irrigation lines. And so even though it's not in the stock plants, it's in the facility. And given a really hot day, many of these Pythium species grow at about 35 centigrade. So it's super hot, but they flourish. And under those conditions, the plants just collapse. So Pythium has its own challenges, even though the inoculum doesn't necessarily originate from the stock plant. It's in the facility and hard to get rid of.
Samir, we went through this with a large greenhouse down in Leamington, and they were irrigating all their cucumber plants in the same manner. And when we tested all the zones where the water was coming up or going out, every pipe was contaminated with Pythium ephinedermatum, which is optimal temperature is 35 degrees Celsius. And when every plant was inoculated, and when the temperature in the greenhouse hit 35, all the cucumbers collapsed. So they went out and spent $3 million to get rid of this irrigation system and put in overhead irrigation, and they haven't had a dead plant since.
I wonder
Yeah, I
too.
mean, that's the extreme control method. But as you know, when we had this heat wave here in BC, the greenhouses were 50 degrees. And you talk about a Pythium outbreak, they must have lost 80% of their plants, no matter what you did. It was so hot that even a slight root infection caused complete mortality. Now that's unusual, but as everybody knows, global warming is going to create way higher greenhouse temperatures. And that's going to create some other disease problems later on.
Samir, I was surprised too that they were using a flood system rather than some sort of trickle system. If you had a trickle system, you wouldn't need to recycle the water because you wouldn't be using nearly so much. Could you and also reduce the capability for infection, and those trickle systems aren't hugely expensive. Why are they still playing with floods?
I guess part of it is just the logistics. Those plants only sit on that bench for 2 weeks. And so, and let's assume there's 100,000 plants. That means somebody physically has to go in and remove the spaghetti tubes or whatever it is that you feed into each block, and manually remove those and then wait for another flow of plants to come through. Right now, it's from a mechanistic point of view, they just set the plants on that bench, and then somebody just turns those nozzles on twice a day and just floods the table. So as you know, with a lot of producers, you can tell them what should be done, and economics or other factors may play into account, and they won't necessarily do it. Now, they're getting away from some of the Pythium issues by adding extra chlorine. So we're recommending chlorination at 5 ppm constant, and that seems to make a difference, except now the whole place smells like a swimming pool. When you walk in, you know they're chlorinating because it smells, but it works if they can keep it at 5 to 6 parts per million.
I had a question.
Sorry, there are some health issues associated with chlorine vapor in a greenhouse. I'm surprised they're allowed to use that. There are people who are hypersensitive to chlorine.
Yeah, now the vents are open and there's hopefully some airflow going through, but being in a cannabis facility for many years, there's a lot of other health issues that I'd be more concerned about, and one of them is the buildup of mold. There's a lot of mold that builds up on product when it's in the dry room, and I've gone in there and sampled with my petri dishes, and boy, those populations got to be able to knock down any healthy human being if you're in that room for several hours a day. So there's definitely many issues to address. I think the chlorine smell is probably not one of the top ones on the list. I think the mold issue is much greater.
There's a couple of questions for Dr. Almagrabi. The first was, what was the species of nematode? You've mentioned it, but they may have missed it.
Okay, the nematodes that we are dealing with is the root lesion nematode, which is Pratylincus penetrans, is the main one for verticillium. Now, when we did soil sampling and testing, of course, we've got other nematodes that were found in the soil, like pin nematode and so on, but these weren't really the ones that are associated with potato early dying.
I think this question applies to your presentation as well. How many cores per acre do you recommend? Do you use a representative sample derived from many subsamples?
It's hard to, well, the way we did it is we took almost one sample every 10 feet or so. So there were a lot of samples using a zigzag pattern. So sometimes it will be 100, sometimes it's 200 cores from the field based on the size. I couldn't tell you really per acre per se, but it was a large number, and your arm gets sore after a while. So.
Can I have a question, because it's not easy to get all you together. So I've been thinking so long about patch management for clubroot disease. Yeah, I'm a clubrooter, as you can imagine, but we are working in fields where we're rotating canola with pulses, different crops. We have different soil-borne diseases here. Have you any idea if, for example, the spatial patterns, the patches that we have for clubroot are like located at the same spots that we have for fusarium or like any other diseases? Because if that's the case, I think that we can go for patch management, including like approaches that can manage broader diseases like soil fumigation, as Mary Ruth mentioned in her talk, or as the anaerobic soil disinfestation. I think it can give us broader perspective, and no, we just manage, because what we usually do is I go and study only clubroot, I go and study only the root rot complex, but we're not getting like an integrated approach for the full system, which is what we have in the Predes.
Bruce may have some comments also, but your one point is correct, that if we're managing clubroot, we're probably managing other soil-borne diseases in the soil as well. Part of the pattern of clubroot in Western Canada is, and Steve Struckoff should be talking about this rather than me. Steve's group did all this work, but because it tends to be spread to a new field on soil on equipment, they always find the most clubroot near the entrance to the right, because that's where the equipment goes first. So that would be different than other diseases in the field, except if those diseases are also brought in on soil from other fields. Where the new virulent pathotypes develop, that could be anywhere in the field probably, because it's selection from the canola crop. In other cases, clubroot tends to be in a low spot in the field, and certainly there could be other plant pathogens there as well. So good point that taking some of these fairly extreme methods like fumigation or solarization should have some other benefits. And I didn't present our data on weeds, but we did show that when you have the totally impermeable film on the soil, it does help to reduce the weeds as well. So there are other benefits for sure.
Just while we're talking about distribution, I thought it was really interesting. I was talking with people that were doing surveys in Manitoba for clubroot, and for in Manitoba, they're not finding the disease in field entrances. They're finding it in low spots and places where water is moving and water is accumulating. And what appears to be happening there is that the main mechanism of introduction into fields is wind-borne inoculum, wind-borne spores that are accumulating at the edges of low spots and causing infection there and spreading out from those wet, highly conducive locations. But as the disease becomes more prevalent in Manitoba, we expect that they'll start to see a lot more movement on equipment from field to field and farmer to farmer. And so it's these diseases aren't static, and where they're present isn't static. So for something like Razoctonia, where the disease has been present for many years in most fields, that they might have a very, very different distribution than where the disease is fairly new, like Aphanomyces, where even though it's moving in the same, likely moving in the same way and finding the same conditions conducive, they may not be present in the same areas. One silver bullet is not going to work for every target.
It will also depend on the biology of the pathogen and what conditions favors its development. So, I mean, clubroot and Aphanomyces are a good example. They have a lot of similarities in terms of what conditions favors them, and so I would assume there's a lot of overlap there. So some of that was already mentioned, right? Sort of depressions where you have a lot of moisture, maybe areas where you have a lot of soil compression, etc. So I wouldn't be surprised if you see a lot of overlap in the patches for those two diseases, simply because both pathogens are favored by those very specific local conditions.
Thank you all.
There's some questions from Tahara in the chat because she's not sure we can hear her, but the first is for Khalil. Well sampling, were those sites chosen randomly from growers' fields or selected by positive disease symptoms?
The ones that I did the survey on were just random. Like, we just went and asked whomever grower is interested in sampling their fields to do so. However, the ones where I did my experiments were selected not randomly. The grower knew that his fields and knew which ones had issues with, and sure enough, when we took soil samples before planting, they had high levels of verticillium, high levels of nematodes. But he could tell you, because from his experience, he knew where the problem is. And that's where I did my plot experiments in the last couple of years on the same farm, but different fields with known issues. Like, visually, he knew, and the yield wasn't as good as it should be. But the survey ones were random, just to see what's going on in New Brunswick, generally speaking. Before 2016, we really didn't know what the problem was, or if there was a problem. But that was an eye-opener, and after that, we started doing more sampling and, revised the way we did it and the timing and so on, and even, plant sampling too.
Khalil, have you ever found a field that had no disease?
I had maybe 3 or 4 that had 0 nematode and 0 verticillium.
And you know what that grower is doing?
Yeah, he's one of the best growers, and he always uses fumigation. But not everybody agrees with that, and not everybody can afford this. He uses chloropicrin, which costs him $600 per acre. The other grower, which I'm dealing with now, he uses Vapam, that costs $390 per acre. But the first grower, instead of using crop insurance, he uses products that do give him a good outcome. And he's one of the best growers in the province, produces high yield, high quality. You go to his farm, it's beautiful. So he's doing the right stuff, and following up with the science and the literature, and he calls me every time he needs. He takes soil samples every year and tests them for diseases and for nutrients and so on. So he's putting a lot of time and effort into it, but he does get the benefit from it at the end of the day.
Thanks. Yeah, I guess that fumigation doesn't replace crop insurance for hailstorms or anything like that, but that's a risk, true. I guess, taking. Here's a question for Mary Ruth and Joji from Tahara. Solarization or anaerobic soil disinfestation. Do you believe this would be a better technique to control other soil-borne pathogens such as nematodes? We can start with Mary Ruth.
I was at a conference on anaerobic soil disinfestation a few years ago in Europe, and I'm trying to remember some of the specifics, but my best guess is yes, but right now I can't think of the data to back that up, so maybe Tahara has more data.
Joji?
So the question is effect of ASD on nematodes?
Yes.
Yes, it's been a lot of literature demonstrating the effectiveness of ASD for nematodes. And as I show you briefly, a lot of Dutch studies and Japanese studies shows the ASD can control nematodes. And when they use the liquid carbon source like ethanol, they can treat the even deeper subsoil level too. In California, fortunately, or unfortunately, we don't have much nematodes damage on strawberries, knock on wood, but we don't have that data. And it seems like temperature is also important to control nematodes by ASD. And the warm region like Florida also have a good control of nematodes by ASD.
Okay, we have a hand up, Sajid.
Yes, thank you. I just want to ask Khalil, because the nematode population, it's very heterogeneous in the field. And if you read the literature, then they show, okay, 20 to 30 cores per acre. But as you mentioned, you were collecting a core after every 10 feet. So in real term, that means in the real world, what is the optimal number of cores? And I don't know, I didn't understand that if you combine your all the cores and you use a representative sample from the acre, or what you do. Yeah, that's my, yeah.
Yes, Sajid, you're right. Now, the larger the number of cores or subsamples from the field, the better representation you get. And grid sampling for those that do soil nutrient testing, they know how that works. I think grid sampling is based on the field size, and they take more soil samples for nutrient analysis. That would be the best time to take soil samples for also disease analysis. So, you know, same concept, same idea. And yes, you do, at the end of the day, I mean, you can test every core if you want to, if you have the money. It's so expensive. Between nematode and verticillium testing, it costs me about $200 per sample. When I do both, you know, PCR and culturing for the verticillium and also identification and number for nematodes, almost $200 per sample. But if you combine them all together from one field, then you get a representative sample, generally speaking. And it's not new really. There is literature that you can find already on sampling of soil for nematodes that's published by nematologists from a while back from Agriculture and Agri-Food Canada. I think Joe Kempinski for Ag Canada's people, they know him. He used to be the nematologist in NPEI. So there's some literature that can be helpful for that, Sajid.
Yeah, and about the sample processing, Khalil, because
yes.
because number of classical nematologists are, I think there are not many, and then I was now sending samples to A&L lab, and they use this real-time PCR, so they have the calibrated they have the curve, and then they know how many, so they extrapolate based on their real-time PCR data. I don't know how exact is it. Because I'm more of a classical approach, so still counting. So that's a little bit tricky. And the university from where I graduated from Wageningen, so developed an assay for 63 species of nematodes. So they have defined the linear equations, and they just extract the DNA from the soil, everything, and then they run those primers, and they are very confident that their assay works.
Yeah, and I think we're working on this too, Sajid, in Canada. Dr. or Benjamin Mimi, I think his name in Quebec. He's a molecular nematologist. He's working on this to see if there's a relationship between what we do with the old Perman method of counting and identification versus what he does find with PCR. Now, what I can tell you with the PCR, at least with the verticillium part of it, I have more faith in the old culture method when it comes to verticillium, where you get the number of microsclerotia per, gram of soil, than the PCR method, because the PCR is qualitative per se, and you don't know whether what you are extracting is active, alive, or not. But when you do get microsclerotia growing on a plate, then you know it is active and it's alive. So that's my take on it. And lately I've been doing both, and we've done this all across Canada. There was only one province that had a good correlation between the PCR results and the culture results. The rest of the samples that came from across the country were not conclusive. You couldn't draw a correlation between them. So we're still working on that aspect of it. But that's an excellent question, though.
No, I think, yeah, I agree with you, because I still believe in counting of nematodes, because metagenomic approaches in case of nematodes, they are not very well developed, and it is hard. And because when I receive the results, for example, if there are 2 nematodes, I'm a bit suspicious actually when low number of nematodes they detect. And so that means I think that sometimes it is like false positive, something like this. Thank you.
You're welcome.
Okay, there's a question directly to me for Tahera. I don't know if Tahera can answer verbally, but maybe in the chat. Has she looked at Phomopsis being involved in ASD disease? There are 2 scientists in the Kentville Research Center working on a national survey for this disease. So yeah, I don't think we can hear you, Tahir. So if you could answer that in the chat, would be good. Question for Zamir: has hydrogen peroxide been used in the hydroponic system and what are the pros and cons?
Yeah, so hydrogen peroxide is registered as a cleaning agent on cannabis, but because it's not very long lasting, in other words, it breaks down pretty fast once it's exposed to UV, it's only used as a surface cleansing agent. So for example, they'll actually take their cuttings before they're actually rooted and dunk them in a solution of 1% hydrogen peroxide just to get them cleaned up on the outside. But it's not used within the hydroponic system itself because it's not long lasting. Less durability than something like chlorine, which will last for a few hours. So it's more or less a surface cleansing agent.
It also causes corrosion, so you know. So it's very corrosive when you use hydrogen peroxide in your system.
Okay, I think we have reached the conclusion of the meeting. Thank you everyone for joining, and I especially thank the speakers for joining us today. It was an excellent session, and good to see all the different work going on across Canada on different soil pathogens. I think there's a lot of work that overlaps here. I also want to thank Amy and Andrea as the co-session organizers and the Canadian Phytopathological Society for hosting the meeting. We did record the meeting. CPS has still got to sort out what to do with the recording, but it'll likely be available to anyone who registered today and potentially others who want to pay to watch it. So with that, have a good day, everyone, and a good weekend. And look forward to more workshops being offered by CPS over the winter.
Thank you, everybody.
Thank you very much.